If we don't act now with passion and commitment, we're never going to get to a world without Alzheimer's.
1 in 3 people all over the world will die from cardiovascular disease. Every single person's lives are touched by cardiovascular disease. We have to come up with novel ways of solving the problem.
As we get into the challenges of more and more precision oncology, smaller and smaller subtypes of cancer, we do have to think about this urgency of making progress.
A new therapeutic age is coming to brain disorders, both neurologic and psychiatric disorders. But I think unless we approach these brain diseases strategically, progress will be way too slow.
This whole messenger RNA technology now being used for the COVID vaccine, I think you can travel to cancer, to personalized or precision medicine in many ways.
We're going to be able to predict when a patient first comes in what combination of drugs to use on that patient based on those massive buckets of data.
I think the last 20 years we've been focused on really reading and interpreting the genome for risk. And I think the next 20 years will be rewriting the genome for health.
What's exciting at this time is we're actually talking about definitive curative therapies, gene therapy and cell therapy. I think we need to have the type of ecosystem that supports that innovation.
Please welcome Richard Sohl, Head of Boston Office and Senior Advisor of Strategic Initiatives, WuXi AppTec, to host Day 2 of the 2022 WuXi Healthcare Forum.
On behalf of WuXi AppTec, I'm thrilled to welcome everyone to the second day of the WuXi Healthcare Forum. This forum is themed Call to Action, and it aims to support the World Health Organization's key initiatives in tackling some of the world's most important health challenges, and in particular the urgent need for intensified global focus in preventing and treating chronic diseases such as cancer, cardiovascular disease, dementia, and other global health challenges. In yesterday's session, we learned of the unique challenges associated with each of these diseases. Our speakers addressed and spoke to their vision of the future and the promising technologies and approaches that will take us across that journey. Today, we wish to take a deeper dive into that innovation that is driving the emerging approaches and treatments and therapies in cardiovascular disease, cancer, neurological disorders. Our panelists are working on the front lines of innovation, and while they come from a wide variety of backgrounds and organizations, they all have one thing in common: a strong commitment to do the right thing for patients. But before we get started, let's welcome Dr. Minzang Chen, Co-Chief Executive Officer of WuXi AppTec, to share his views on healthcare innovation.
Please welcome Mingjing Chen, Co-CEO, WuXi AppTec, for his opening remarks.
Thanks, Rich, for your kind introduction. Welcome, friends and colleagues. It's a great honor to join so many distinguished innovators here today. Each of us shares a passion for scientific innovation and for doing the right thing for patients. The challenges we've been discussing throughout the WuXi Healthcare Forum are significant, and I continue to be inspired by the heroic efforts from our global innovation ecosystem in facing them head-on. It's a great honor for WuXi AppTec to partner with so many frontline innovators to deliver groundbreaking treatments to patients. Innovation is a key to addressing the challenges we face. Promising new technologies, such as new modalities, manufacturing technologies, and novel delivery systems have the potential to revolutionize healthcare. But the success, particularly in our industry, really comes on the first attempt. We must remain committed to our work and view each challenge as motivation to think more creatively about how we can achieve our goals. The innovators you will hear from today and tomorrow embody this spirit, relentlessly exploring new frontiers in their daily work. How to move the needle in neuroscience R&D? How will emerging technologies today transform cardiovascular and cancer care tomorrow? What will healthcare look like in 2030? These are just a few of questions we will be discussing for the remaining of the conference. As you all know, global solutions require global collaborations. Many of our speakers at the forum are the pioneers in the healthcare industry, leading the way in the development of new medicines and healthcare solutions with both creative thinking and actionable approaches. They exemplify how a diverse ecosystem of collaborators work together to deliver scientific and medical innovations to patients. WuXi AppTec continues to be inspired by these frontline innovators who are devoted to rising to meet this challenge in our own work. Over the past 20 years, we have been diligently building our global enabling platform and proudly supporting our partners with our unique CRDMO and CTDMO models. Our vision is a future where every drug can be made and every disease can be treated. I hope this is a future everyone attending this conference is also looking forward to. I invite everyone to join me in renewing our commitments to advancing better medicines faster for patients and building a healthier future for all. Thank you.
The record speed in delivering vaccines and therapeutics during the COVID pandemic exemplified our industry's ability to collaboratively tackle pressing healthcare challenges. How can we continue to learn from and be inspired by what's happened over the last several years to create an ecosystem that accelerates innovation for chronic diseases, preempts illness, and makes healthcare solutions accessible to all? Please welcome Stephen Hahn, CEO Partner, Flagship Pioneering, and CEO of Harbinger Health.
So first of all, I'd like to begin by thanking the WuXi Healthcare Forum and organizers for this opportunity to speak with all of you. I'm really pleased to be here, and I was pleased when the organizing committee told me that the conference would focus on how technology will shape the future of health by predicting, preventing, and personalizing treatment and care. This is such an important topic, and it's where the private sector and innovation in medical products can really help humankind. Now, we live in extraordinary times, and we're really fortunate to be part of this. We're witnessing an extraordinary amount of progress against human disease. I mean, I think back to when I started as a physician, and some of the things we're talking about in terms of being able to address problems that we're able to address, treatments that we're able to give, really just remarkable and extraordinary. And we are making progress, and I think notwithstanding our experience with COVID, we continue to make that progress. And this progress is the result of investments in science and medicine, which are allowing us to develop new treatments for chronic, life-threatening, non-communicable diseases like cancer, my specialty, diabetes, and also, of course, cardiovascular disease. But there are many other diseases that we never even contemplated that we would be able to treat, and now we're thinking about those problems and those diseases. And I'm going to tell you why I believe we have every reason to be optimistic about what lies ahead. Make no mistake, it'll be challenging to realize this vision of advancing innovation and helping humankind with the many diseases that face us. The global SARS-CoV-2 pandemic has and will raise in the future continuing obstacles. But if we resist the temptation to retreat to our pre-pandemic safety zones and continually to actively foster an environment that lets science and research and innovation flourish, I believe that technology and biology will transform medicine. Frankly, they'll transform our lives. And more importantly, I believe these advances will make health equitable in ways we thought not possible in our lifetime. And I just want to stop there for a moment because this really is in the hands of people at this conference. What is possible for us in the future will come from the great work that you're doing. And I do believe that based upon and building upon what we've experienced during the SARS-CoV-2 pandemic, we'll be able to really have a tremendous and bright future. So what shapes my thinking? Well, from December 29th December 2019 through January of 2021, I had the privilege and honor of serving our country as the 24th Commissioner of Food and Drugs. And as commissioner, I established, interestingly enough, when I first got there pre-COVID, 3 priorities for my tenure. The first was to spur and enable innovation, which really means getting innovative medical new products into the hands of the American people and American product providers as safely and as quickly as possible. The second was unleashing the power of data, and in many circumstances, real-world evidence to make better decisions about the safety and efficacy of medical products. And I'm going to talk about this a little bit more because I do think this goes hand in hand with our ability to spur and accelerate innovation. And finally, incorporating the voice of the American consumer and patient in all the decision-making that was made at the FDA. But that holds for the private sector as well, as we develop innovative medical products. Now, what I didn't realize when I started at the FDA in late December 2019 was that in 6 very short weeks, with the emergence of COVID-19, nothing would be more important to the American people and the world than enabling innovation from the private sector. That is a lot of people coming together and collaborating, and the private sector helping us to get to a place where we need to address an urgent clinical problem. And there are more urgent clinical and health-related problems and other problems than just SARS-CoV-2, as I'll talk about. Few of us could ever have predicted such a timetable, particularly because the normal timetable for vaccine development is counted in years, not months. More than anything, these previously unimaginable achievements underscore the importance of a vibrant, robust private sector, as I've said, that deploys its resources to create innovative, life-changing products. That's where you all come in. That's where the partnership and collaborate begins. So the private sector, of course, was substantially aided by investment in basic research by the US government, and also the terrific work of our academic colleagues in the academic community, where I come from prior to FDA and prior to my current position. And given what was accomplished in medical innovation in response to the COVID-19 pandemic, let me posit this question to you. Is there any reason why we can't expedite the development of medical products in a similar timeframe? So maybe it's not 11 months for a vaccine from identification of a virus to an authorization, but why not? I believe that by creating and sustaining an environment that supports the private sector with appropriate oversight, we need that oversight with respect to safety and efficacy, we can see more innovation and even faster development cycles. And isn't that what it's all about? More quickly getting innovative products into the hands of providers, patients, and consumers. Now, we saw this impact, the impact of enablement and the power of collaboration during COVID-19 pandemic. Now, if you think about what happened during the COVID-19 pandemic, authorization occurred oftentimes on more limited data sets by necessity to get products into the market. And what the agency and sponsors did during this time is look at the performance of those products in the real world setting during the pandemic and use that information to feed back. That sort of approach is used in accelerated approval and other unique pathways at the agency. And my question again is, why can't we do more of that? As long as we ensure the safety and efficacy of the products, and as long as we come back and we can revise our decision. Let's talk about how we can use intermediate and surrogate biomarkers to actually accelerate approval. And then let's do the job in the private sector of real world evidence collection. So in real time, we can address how these products work and then change the label and the marketing associated with this based upon what we learn in that setting. We've done that in the cancer sphere for many years. Why can't we do that for all other diseases? And why can't we make that time frame shorter? So we have to change the mindset associated with this, and we have to reduce the obstacles to that. A lot of smart people, smarter than I, can get in a room and think about how this would work. But people are dying every day from these diseases, not just cancer. And it is an imperative for us to actually think about ways to accelerate innovation. I think we've seen some pretty significant increases in drug approvals, and that's really good, and also medical products, but it's not good enough. And the minute we say that it is good enough, we've kind of lost the battle. Maybe the war, who knows? But the point is we have to always do better. And we have to set really lofty goals for this, achievable but lofty goals. because people's lives are at stake. People are depending upon us to do it. That's going to require a different mindset for the private sector, but also a different regulatory mindset as well. And those are the things that have to come together. I think on the drug developer side, really have to think about innovative ways to use those technologies to better refine our approach to the development of drugs, and particularly the clinical development of drugs. So how when you develop drugs, do you define people with diseases as opposed to illnesses? Meaning, how do you identify those people who will benefit the most from the intervention? And that means understanding the biology and the data associated with that. Now, that may be the use of a biomarker. That may be the use of a constellation of clinical symptoms or specific ways to establish a diagnosis. But we have to get better at taking a much more directed view for the people who will most benefit from the therapeutics that we're developing. So I say, let's really go in heavy on the accelerated approval pathway. Let's decide the areas that are most urgent for our country and for the health needs of Americans in the globe. And let's put in place the parameters around which we can provide a very robust regulatory oversight. And I think the nexus of that is not only the data and the analytics, but the biomarkers associated with real disease endpoints that matter to people. Now, I want to now focus on another issue, and that is we've learned a lot about pandemic response, how we need to improve our response, and also the health challenges facing us. And all of you are working so hard to address the challenges in human health that are there. In my opinion, the biggest ones, the biggest healthcare challenges are related to chronic disease. Some people refer to these as the slow pandemics. And of course, not only the chronic disease, but threats of future pathogen-related disease. And the major reason that I joined my current position at Flagship Pioneering and Harbinger Health after my tenure at FDA is that we're asking provocative questions with a long-term view of human health. What if we could prevent disease before people get sick? Can we build a world freer from disease than at any other point in human history? Can every person be as healthy as they possibly can be so that society and individuals flourish together and equally? And it's very important in this setting that we reconceptualize our entire approach, in my opinion, to focus more on preempting illness than simply treating it when it arrives, whether it's a novel infectious disease or chronic disease. And this is fundamentally a shift in how we think about healthcare. And I come to you as a person who spent 35 plus years in healthcare and academic healthcare. But I do, and I admit to you that we treat illness now. We don't really preempt it the way that we should. But we're living in a new era. Frankly, I'm just as concerned about the threats associated with antimicrobial resistance, just as an example. But the others are already here: cardiovascular disease, diabetes, obesity, cancer. And these non-communicable diseases kill 41 million people each year, equivalent to 71% of all deaths globally. And the United Nations, as part of its Sustainable Development Goals, wants to, by 2030, reduce by a third the mortality rates from non-communicable diseases. I would like to see us achieve that goal. And I believe that biology and technology can help us get there. Now think about this for a moment. To achieve the goal of preemption, and I hope you all will put time into this, we will need to rethink about how we approach clinical development and also the regulatory framework. It took us decades to identify the risk factors, biomarkers, if you will, for cardiovascular disease, cholesterol, hypertension, diabetes, and then to develop interventions. We cannot wait 50 years to solve the problem of preemption in chronic disease. We have to think about how we use new ways of analyzing and using data to actually develop medical products, AI, ML, other models with existing data. The information, much of it is out there, that allow us to help what we call build a global pathogen shield and identify risk factors and biomarkers for pre-disease so that we can intervene when the threat of that chronic disease is down the road. Again, we're going to have to reconceptualize this, but let's think about this with a current paradigm. How do we take all the discovery tools and information technologies that could spot and monitor actual potential pathogen outbreaks or markers of pre-disease and then intervene, similar to how we use our sophisticated and extremely valuable National Hurricane Center. We can do this. We just need to set up the framework to be able to do it. We call it, where I work, intelligence for pathogen preparedness, but also intelligence for identification and intervention for chronic disease. So rapid response won't be enough. We need to go beyond that and be pre-armed with the necessary tools. And that means building the infrastructure around information, data analysis, sharing of data, and then in a preemptive sense, thinking about what sort of therapeutic and preventative tools can be developed so that if a threat does emerge, we can very rapidly develop those and have a cycle time that's measured in months, not years. If we're able to model what happens with pathogens, AMR, for example, and we're able then to design therapeutics for those particular pathogens and how they mutate and how they evolve over time, we can be proactive and we can actually have those therapeutics developed or at least conceptualized before the pathogen actually arrives. Now, we have to have confidence in the fact that these predictive capabilities actually are able to predict what happens and what will happen in the future. But I'm extremely confident that that is the way that we're going to move forward. And I am very confident that that's how we will accelerate innovation. So, I say our goal should be, if there is another, and I think there will be another pathogen that's identified in the future that could affect humankind, let's have a vaccine and monoclonal antibodies in 3 months, not 11. And what are the predictive capabilities in the platform to build this pathogen shield so that we can actually get to that point, so that we have something that we can, if you will, immediately put into the clinic and preclinical studies and then accelerate the innovation. So I think it lies on the data and innovation side. And I know at Flagship Pioneering, we're leaning in heavily on our AI and ML and data analytic capabilities to actually make interesting observations that can help with the development of innovative medical products. And I want to end on this. What excites me the most about these new approaches is the opportunity, and I will say the moral imperative before us, to make new innovations in healthcare more accessible to all. And I think it's incumbent on all of us in the private sector to ensure that anyone can have access to the latest solutions and that no one is left behind. We've seen what happens when people are left behind, and there is no reason why each of us, regardless of cultural or socioeconomic status, shouldn't be able to walk into a local pharmacy or go online and take a simple, inexpensive blood test to better understand their health outlook, giving them the opportunity to act earlier. By breaking open the existing disparities in our system, we can tackle them. And by embracing new scientific approaches, especially ones that prioritize more effective prevention and earlier diagnosis, we can change the way we approach health for everyone. Think about this future, a future where healthcare is distributed, where all individuals have access to tools and information they need to make decisions about their health. And the focus is on longitudinal wellness, not just treating an acute disease. Let's create the regulatory paradigms and the private sector infrastructure to aid in solving the problems to access. I am more confident than ever that we will continue to learn from and be inspired by what's happened over the last several years to create a new kind of health innovation ecosystem, one that is primed to accelerate the development of life-saving diagnostic approaches and treatments to ensure healthier and more productive lives for everyone. You all are at the forefront of this new world, and I am confident that we together can make this happen. Thank you.
Facing the dire need to provide therapies to stop, slow down, or prevent neurological disorders, and neurodegeneration in particular, what is holding us back? Are we on the verge of a breakthrough? How can we accelerate this endeavor? Please welcome Lawrence Barker, Partner, the Dementia Discovery Fund, Christina Isaacson, Partner, Lightstone Ventures, Philip Heltinch, Head, Dementia Fund, EQT Life Sciences, and Rana Al-Halak, Partner, Pfizer Ventures.
So welcome to the WuXi Healthcare Conference. It's my pleasure to invite or to welcome Lawrence, Christina, and Philip here with me to discuss innovations in the neuroscience space. I would like to kick off first by inviting you each to maybe give a little bit on where you are now, what you're working on. I did just want to highlight that this is a CNS forum, and we are going to be focusing on neurodegeneration, maybe less so on the mental health psychiatry aspect. Of course, please feel free to jump in with those, but I just wanted for the audience to know that we may have a little bit more of a focus on the neurodegeneration, not to say that the mental health is not an area of innovation and of a severe unmet need, but that is the current focus. So maybe we'll start off with Lawrence. Would you like to give us a quick intro?
Thanks, Rana. Pleasure to be here today. Lawrence Parker, I'm a partner with the Dementia Discovery Fund. We're a venture fund really set up to stimulate new treatments for neurodegenerative diseases that cause dementia. And we've been, over the last few years, building an early stage portfolio, which we're quite excited about. And I hope through the course of this discussion can tell you more about that and how we're trying to positively impact dementia for the benefit of patients.
Christina?
Great, thanks, Rana. Christina Isaacson, I'm thrilled to be here today with this esteemed panel. I am a partner at Lightstone Ventures. We are a fund that is really focused on supporting and building new companies across a number of therapeutic areas, including neuroscience. I am actually a neuroscientist by training and background, and have spent quite a bit of time in different operating roles, mostly from a business perspective, but have never lost my passion and commitment to bringing forward transformational therapies for CNS disorders. At Lightstone, we have made several investments in the CNS space. While we do look at other therapeutic areas, it is an area that we are very excited about. We've just raised a third fund of $375 million and hope to put that to work, including in the CNS space.
Great, thank you.
Very thrilled to be here today, and it's always exciting to talk about this specific topic, and especially in with such esteemed colleagues. I'm a professor of neurology at the Amsterdam University Medical Centers. I spent 30 years of my life working on biomarkers and diagnosis, improving diagnosis for neurodegenerative diseases, specifically dementia. And since 1.5 years, I'm also the managing partner of a new dementia fund, an investment fund under the umbrella of Life Science Partners, now EQT. And now I do this for 80% of my time with a team of 5 people. We're actually operating in exactly the same space as Lawrence is. We are a little bit later in the sense that we do not real fund discoveries, but probably a little bit later, phase 1, phase 2 studies, but in the broader neurodegenerative space with anything that has to do with dementia and neurodegeneration, we look at it.
Great, thank you. So just for completeness, I'm Rena with Pfizer Ventures, and we are active investors in the neuroscience space as well as other areas. I would like to kick it off. I think we all appreciate that neuroscience and neurodegeneration is an area of unmet need, and obviously we're all here to try to make that difference and to bring therapeutics to patients. I would love to hear for us to talk a little bit about the magnitude of the problem and where we've seen progress. And then I think maybe we'll then kick off our discussion around what we can do, to move things forward. But just starting with that magnitude of the problem, and maybe, and then I'd like to maybe turn it over to Philip. Give us your thoughts on how you see this and how you see the field currently, and where we've come and what we have yet to do.
Yeah, so I'll try to be brief, but I think the magnitude of problem cannot be overstated because it's really huge. If we only focus on diseases with dementia, and all of them are actually neurodegenerative in nature, the WHO has calculated that there are roughly 43 million people suffering worldwide, and that number is probably going to triple in the next 30 years if we don't do anything. And that sort of numbers are always a bit there are many diseases that are very prevalent, but the problem with dementia is that it makes people very dependent on others. So the magnitude of the problem by itself is actually exaggerated by the fact that 3 times as much people have to care for these patients. And it's also a very costly disease. So if we are, the three of us or the four of us, are not able to come up with solutions very soon, the healthcare systems will basically collapse and will not be able to take care of all these patients. So we are in dire need of providing therapies, especially to stop, to slow down, or to actually prevent all these diseases that we are discussing. And I joined sort of the Dementia Fund because of that, because working in this field for so long time, I've seen a huge progress in making the diagnosis, in finding biomarkers, understanding the disease, the underlying pathology, so much better than we used to 10, 15 years ago. But still, we are in the very early days in terms of developing good therapies. So that's why I think the only solution I see is that we join forces together with academia and big pharma and the venture capitalists in between to make this happen. And I think there are enough opportunities to do this.
That's great. Christina, maybe you could talk a little bit about ways that we have been trying to move forward. I think, has the field changed in its approach, and maybe elaborate on anything you'd like as well, based on what Philip just mentioned in terms of the magnitude of the problem and how we're going to tackle this?
Yeah, I think we've seen, significant progress, I'll say over the last decade in particular. And I think there's more progress, though, to be had, right? So I think we need to acknowledge that. And I think in large part, you're hearing from folks who are committed and, want to invest in the science and the technology and the clinical understanding, right, of bringing innovation forward in this space. But we need more of that. I mean, I think that's fair to say. The improvements we've seen, I think, have been largely, Philip just referenced a few of these, better understanding and identification of biomarkers that have enabled, I think, some opportunities in clinical trial designs. I think the part that I'm, really excited about is that some of the science and biology and pathophysiology understanding is now opening up new targets, right? New areas of biology to pursue, so we can move away from, certain hypotheses, around protein aggregation that have been, really explored, I think, quite extensively. And unfortunately, other areas have been a little bit more ignored. And I think now you're seeing that those other areas of biology are now coming up. So to name a few, for example, the role of lipids in neurodegenerative disease. I think lipid biology is a very an area that's well understood and known. And I think now we can apply some good knowledge and good basics to the neuroscience space. So I'm very excited about that. And then I'll conclude with an example. I think we're also seeing some kind of pages out of the oncology playbook of trying to move to more personalized medicine approaches in CNS. And I have a good example with a recent investment we made in Vigil Neuroscience. This is a company that is very focused and committed to the role of microglia in neurodegenerative diseases. And the company's lead programs are focused on a novel target that's been identified called TREM2, which is specifically expressed on microglia. Now, the company has also identified a unique clinical strategy to really exploit and understand the role that target plays in neurodegenerative disease. And they're pursuing a rare adult onset leukoencephalopathy that was only identified really 12 years ago. It has a lot of overlapping hallmarks with a broader set of neurodegenerative diseases. And so they're actually in the clinic pursuing that very quickly and aggressively. And the hope is that by studying a very well-defined, genetically defined population like ALSP, that they can expand into broader sets of neurodegenerative diseases from there. So I think we're also starting to see those strategies that we commonly see in other therapeutic areas move into the neuroscience space.
That's great. Lawrence, I think I'd love to hear from you, given the diversity of the portfolio there. And I think this precision medicine approach, your thoughts on that?
Thanks, Ryan. Just picking up on two of these themes and not referring to the problem. I think Philip laid that out, very plainly and clearly for all of us. But what is changing? I think I like to view this through two lenses, and they go hand in hand. This diversification this deeper understanding of biology, which is now supporting a greater variety of approaches that are being taken. And I recently was privileged to hear a number of presentations around sort of overviewing. Let's take the Alzheimer's pipeline as an example, where now we're seeing the lion's share of new programs entering the clinic are basically against new targets, new approaches, outside of some of the paradigms that perhaps have been previously the focus. And I think that's a wonderful indication that actually not only are we seeing that at the very early stages of unlocking new targets in biology, but that's beginning to be translated through into the patient setting. Now, yes, I agree with everything. We've got a long way to go, but that is happening more widely and more deeply. And we see that across our portfolio with a range of different mechanisms now moving into the clinic for some of these diseases. So that I think is the real positive. And the approaches that often people start with when looking to exemplify a certain biology is to use this precision medicine approach, right? Use a genetic subset of better characterized, understood patients as a starting point before you then expand out into a broader sporadic population, which of course from an unmet medical need perspective is where we must end up ultimately. And I think my second point relates to that and something we must always keep an eye on, and that is of course the capital to enable these really important studies. And I think what we're seeing is positive trends, even just the last 3 or 4 years of additional capital coming into the space, building on the fact that we have greater validation, cautiously put, in some of these areas. And we are seeing an expansion of capital available, whether that's across, venture, across the broader pharma industry, beginning to lean back in. And I think the key word I would use here is partnership. Doing so in strong collaboration with, Philip, you mentioned key academic centers. I think that's absolutely true. To close it with venture. And even of late, if I reflect back over a portfolio that I know best, our own, we're seeing greater pharma deal making happen as they start to want to lean in. But it is frequently focused on this precision medicine approach. So that is resonating, but I still think, if you looked at the deal making or the level of activity versus the field of oncology or others, we're still a fraction, but I think we are making really, I think, exciting progress in the right direction.
I would just, it's a very boring session actually, because I completely agree with everything that has been said. It's so well put actually. And I would just remind us, for probably the audience, it's good to be remembered that we are in the field of neurodegeneration is where oncology was 10, 15 years ago. Yeah. And that means that we can make the same progress if we all put our shoulders onto it, and especially also the capital to do it, because that's, of course, the magic in the whole oncology has been the research and the capital and the demand, the needs that the patients actually voiced. And this is now also happening luckily also in dementia. There is more active organizations that are actually putting us, saying you have to work harder, and also to the authorities, and you have to sort of get drugs earlier to the market. I think a very important example is, of course, Christina carefully avoided the word amyloids, but that's still the main thing. But so Aduhelm, the approval, whatever you can say about it, the pros and cons, but the fact that it happened on the basis of biomarkers is already a big achievement, and it's opening the door, and we have seen this all happening, actually, opening the door to many other treatments, also on amyloid, but also and specifically outside of that amyloid, because everybody realizes there will be no single silver bullet that actually helps this problem. So we need all the approaches, all the shots on goal that we can do. And that's why we need many more of the funds that Lawrence and I are involved in. We need many more big pharma actually to go along with us, and many more researchers, and that's sometimes forgotten. There are still 40 times less people working in neurodegenerative research than there are in oncology research, and that needs to change. So we have to also inspire our younger colleagues to go in this specific field and do research on it. Otherwise, it will not work.
Yeah, Philip, maybe can I pick up on that thread? I think that's a very important point. I was just thinking a very similar thought, and so maybe to add to the boring nature of this panel, just kidding. I think it's an important thing, because the other piece that, I think the oncology space is in quite well is kind of the cross-disciplinary collaboration as well. And Lawrence, this is your point on partnership. I think there are different forms of partnership. But I think, neuroscience is, yes, a field of expertise, but we all know that the immune system is playing a massive role in the establishment and progression of many neurodegenerative diseases. And, the broader, field of neuroinflammation is something that I think is well accepted. That is, it is a key aspect of what we're all studying and the biology that we think is really important. But I think the more collaboration we can see across disciplinary areas as well, I think the more we'll learn, and the faster we'll make that progress too.
Yeah. Perhaps just to take that theme one step further, what I'm seeing frequently is with companies that have, let's say a novel mechanism they're studying preclinically, and they're trying to think about how to get the fastest, most robust clinical demonstration, of benefit using that mechanism. And that's not always to go head on into the neurodegenerative disease first time. Some do, yes, particularly where the translational plan is perhaps, everyone feels we understand the nature of the biology sufficiently to do that. But frequently, this is to step off into some peripheral disease or genetic subset, but still maybe not central, to demonstrate mechanistically, and then take a central approach. And I think that's both good insofar as sometimes that can be done more quickly, but I think the caveat is, how well does that really represent our understanding of the mechanism if, for example, you were to study peripherally versus centrally. But I am seeing that approach, and sometimes I think we oversimplify the translation of one to the other, and that's a caution in my book. But it is certainly something we get pitched frequently. So it's certainly something that I think the broader industry is doing as an approach.
And how do we progress that? So I think that some of it has to do with biomarkers, being able to link, two systems and be able to read kind of across the systems with biomarkers. I guess what else can we do to facilitate some of these efficiencies that we're trying to create in the system, right? We're trying to get these therapeutics to patients sooner, and we're trying to find ways to do that. But what else can we do? What's holding us back from being able to do it more quickly to show that proof of concept? Biomarker approvals are obviously are a key point. Anything else that we could think about?
So from my point of view, I think the biomarkers are really very useful to identify target engagement and also early signs of efficacy. And also, if there are no signs of efficacy in early phase, just don't go through. So just stop it. So it helps us probably also, if you look back in the past, many of the drugs that have been gone into phase 3 could have been stopped earlier if we had the proper biomarkers or actually had looked at the biomarkers. So I think that's helpful, especially in the earlier phase of development. Biomarkers are really important, but ultimately we want to do something for our patients, and that needs to be measured. And the problem in the field, as I see it, especially from the Alzheimer, but also the other neurodegenerative spaces, is the readouts clinically are very poor. They are very insensitive to change. They are really not always clinically meaningful, and we don't do a good job at that point. So we have to, as the field progresses, we can use the biomarkers to develop the drugs, but ultimately we need the very sensitive readouts that mean something for the patients as well, to ultimately say, yes, this drug is good enough for the patient. So I think that's a lot of improvement has to be made.
And is that digital readouts, or
can be digital? Yeah, can be. There's a lot of interest, for instance, in all cognitive tests that can be done digitally, even at home. Yes, actually the main point is probably to reduce the variability, because that has killed many of the drugs, because many of the tests are so variable. Depends on the day when you take it and the mood that the patient is in on the morning versus the afternoon. So there are huge differences and huge variability that has to overcome. We showed in a paper just last year that many of the effects that we're trying to see are already there if you just look at the natural progression. And so if you don't treat anybody, you have, by the nature of natural progression or natural variability in progression, you also see changes on cognitive tests that we could attribute to medication, which is not true actually. So we really need to do a better job there.
Just picking up on that, Philip, using if you like, not just traditional measures, but perhaps in combination with some of these digital approaches, looking at different types of measures, not just endpoints, but measures. Is that also an opportunity potentially to look earlier in disease, because we are not, just focused on, a few, but actually a broader range of the types of things we're measuring, right across behaviors and other areas too, that allows us potentially to see changes in people prior to what we're picking up traditionally now, and I'd been able to identify them as having, dementia X, Y, or Z.
Yeah. No, I agree. Yeah. So several sort of wearables, like looking at gait, or perhaps looking at speech, looking at language production, the variability in language production. There are real sort of good examples of other things that we should probably use in an earlier stage. Yes.
Yeah, I've also seen some around kind of eye movement as well. Ability to hear certain sounds, monitoring kind of hearing. It's an interesting area, Ren. I think we're all learning as we speak, I think, in the digital space and how that's going to impact neuro. I will say, though, I think that is one of the spaces where we see more opportunities of applying digital measurements and digital health in the neuro space compared to any other therapeutic area. I don't know if that's true for you guys as well, but I think it's kind of interesting that that's the cutting edge space that people are moving into. And there's a reason for it, I think, just in terms of accessibility of what you can measure. But what I like about it is that it does put the patient in the control seat, right? Of really participate, or their family members. I should also acknowledge that, because I think that's a big part of this, where they're providing the data sets and the input, which I think is really valuable.
Yeah, no, I totally agree, and I think that's, and I can understand if you say that this is probably the most needed in the neuroscience field, because whatever we can do with biomarkers, there's still what we have to prove is that the change in the biomarkers is also sort of reflecting in a change in function or cognition or behavior. And that's the thing that we need to measure better than we are. And in oncology, in my mind, but I'm not an oncologist, I mean, it's always been good if you can reduce the tumor by 50 or 60%, that's already okay, regardless of whether the patient has less pain or whatever. But here in this space, even 80% of amyloid removal doesn't really convince everybody that this is the treatment that we should give if there is no clinical effect.
I think it would be helpful, since we kind of drew the parallel between neurodegeneration and oncology, the two fields. It would be helpful maybe to kind of talk about how they're different. What are the challenges that we see in neurodegeneration that we may not have to, deal with, or don't hold us back as much on the oncology side?
Yeah, it's a great question, Rana, actually. So there are some unique features I think that we should acknowledge as to why this might also feel a little bit harder, right? I mean, I think one challenge that drug developers have is that the brain is a privileged site. We have to deal with the blood-brain barrier, and being able to find drugs and modalities that will be able to access, right, the brain. And I think people have been very innovative, creative, novel to try to tackle that. You now see antibodies in development, right, for CNS-based diseases. And it's really about how much is enough, right, to be able to drug and get an effect. So I think we're taking more risks there. We are, trying to break that forward. But I would say that is one unique feature that we have to deal with in neuro if you want to access the brain.
I agree, Chris. It's stepping off from that, and something that we almost jokingly refer to because it's so obvious, but it isn't, is you can't biopsy the brain in the same way. Obviously, we can take CSF samples, sure, but even my physiology would suggest that's not the same thing. And so we have that problem, right, in terms of how we understand explicitly, or specifically, perhaps better put, what this patient versus that patient might have in terms of doing analysis. And so, interested in people's reflections on that, on how we get round it. You have to find other means and mechanisms to get that level of information which we would dearly love to have, I'm sure.
No, I couldn't agree more. I mean, those are very, very important barriers. And on the other hand, I think also in the general public sense, many of the diseases are still considered to be age-related, and say, why would you bother, because it's just age-related. So there's a lot of awareness and a lot of sort of the identification that we are dealing with fatal diseases that need to be treated as fatal diseases as we do with oncology. That's, for me, is prior and really the most important part.
So to that point, so patients are obviously waiting for treatments and cures and help in any way. What do you see coming? What gets you excited when you think about where the field is, something that's kind of on the horizon? And it's not necessarily a specific therapeutic. It could, also be the regulatory environment, or like we were talking about the biomarkers, anything. Where do you think is going to take us kind of to be able to leap forward in our field, and hopefully, get to where oncology is today?
I mean, just 2 points from me. I mean, the obvious one is, we need a level of clear success from a clinical study in one of these core diseases, using the precision medicine, using the patient subset, which clearly demonstrates benefit, right? Benefit on the key endpoints that people know and understand. And it sounds like such a simple statement, but of course it's not. And unfortunately, we have seen some studies not play out positively, not benefit patients, which have been, I think, a surprise to the field, because they have gone after patient subsets using approaches we think we understand. So to me, even that is not a slam dunk and obvious, but I think that is something which I'm glass half full. And I think we will see in the very near future. There are enough studies with very thoughtful people driving them. We will see some of that success, and that will encourage other people to continue and expand beyond that. So I just wanted to focus on that, because we haven't seen that level of success, I think, even more recently where we might have expected it to. So that's something that I'm seeing from some of the mid-late stage clinical studies, where I think the odds are beginning to stack in our favor, but let's wait to see the data.
I think the part I'm encouraged about, Rena, is going back to something Lawrence mentioned earlier. I think the level of commitment and collaboration that we still see in the space, and that we're starting to see grow, I think is really, really important. Depending on who you are, where we sit in that collaboration ecosystem, we all have different risk tolerances, but it takes all of us to work together to kind of drive and de-risk to every level, so that each of us can play our role in that. And I want to emphasize, I think the academic setting is playing a huge role here, and actually NIH funding is playing a huge role. The more that we can see NIH participate in really supporting, yes, basic research, but even the translation to the clinic. I am hearing more and more of that kind of activity, which is huge, right? Because that is a massive de-risking effort that the academics and government take on, for then some of us to be able to come in and say, okay, we're going to really drive this forward to the access point for patients. So if we can see more of that as well, I think it will be huge. I mean, there are modalities that we mentioned, gene therapy, there's cell therapy, right? That are the potential, Rena, you said the word cure. Those are the potential cures for some of these diseases, but there is a risk associated with them that some of us can take and some of us can't. And so the more we can collaborate, the more we'll see some of that innovation come forward. And I think that's happening.
Yeah. I would just add, I totally agree. I was very happy to see many governments in the world, not only the Dutch, especially the UK government, I really applaud, and also the Australian governments and many others, really putting dementia and neurodegeneration actually first and foremost on the list of things to invest in. And I think that's key, and we need those, and NIH is a very good example. The US government actually tripled the budget, I think, something that's actually needed. And still, it's less compared to the oncology field, but it's a beginning, and we have to do this. And then I think I'm the most optimistic also about the development of biomarkers. So 5 years ago, if you would have asked me whether we would have a blood-based biomarker for amyloid or tau, I would actually laughed. And now it's reality. So it's actually working. We saw at ADPD a new PET biomarker for alpha-synuclein that's actually amazingly doing well in the cerebellum for MSA. So there's a huge sort of development in terms of making biopsies in vivo that will enable us to develop better drugs. So I'm really optimistic in that sense.
And I think there are groups that are very active in that space and trying to be the ones to be the focal points to bring that data together. And so maybe just to that point, the one thing that I think we haven't touched on a little bit in that environment of all of the different players is patient groups and foundations, and just the importance of those, especially as we're going with these smaller diseases that are less known, or we don't understand them as well, or the patients are not there aren't as many patients out there for us to identify and diagnose properly.
Sorry, Philip, just one quick comment from me. I agree completely. And I think, it's on us, it's on the industry to better engage with those groups. And this is explaining better what approach X or Y is trying to do, what the potential benefit of it is. I think we have to much earlier and much more robustly build those relationships so they understand, because we or the actual potential drug will suffer. We will not be able to recruit for these studies in a timely manner. I think it's one of the largest risks currently in the space, is being able to recruit and be able to keep these studies moving forward reasonably. And so I think starting that much earlier than perhaps we have done historically. Also, therefore, to have a prior relationship with patients for whom such an approach might actually be applicable. So yeah, agree. And there's lots of disease area foundations one can do that starting through and in close partnership with.
This is a very important theme, Rena. I'm really glad that you mentioned this. I think I mentioned this adult onset leukoencephalopathy earlier, ALSP. I mean, again, this is a newly identified disease that was just identified about 12 years ago. This is the form that has axonal spheroids, that's the SP part, and pigmented glia, that's the SP part of ALSP. And there's already patient groups formed around this. With a group called Sisters Hope Foundation, which Vigil Neuroscience is part of their, deliberate clinical strategy here. They have partnered very closely with Sisters Hope, and they are a key part of enabling and making sure that, as much as possible, they are successful in driving home a therapeutic for those patients. So I think it's incredibly important to do it as early as possible. And I think the message here is more to tell the patients and their families, their voice matters. And in fact, it's incredibly supportive and helpful to what these companies are trying to do.
That's great. I think we have just maybe a couple of minutes left. I would love to just go around and hear from you. Anything that you would like to highlight that's hopeful, that you're anticipating? We really are on the verge of this wave in neuroscience and neurodegeneration work that's, I think, is going to be fruitful, I think, hopefully in the very near future. So just curious, maybe you could highlight one or two areas or thoughts even. Lawrence, do you want to start?
Yeah, no, sure, Rana, thank you. So I suppose the discussion we've had so far has been really talking about new therapies and the promise that I think they bring. I think the fascinating next phase of that, when we hopefully see success maybe with a monotherapy, is to think then obviously about combinations. This is obviously these diseases, and I think some of it understood than others, are clearly multifactorial. They go through different stages. We are going to need different medicines probably, whether simultaneously, whether at different stages. And I think that's the next bastion. And I think we obviously, we need some success early on with individual approaches to understand actually how you might ultimately end up wanting to use them as a physician with patients. But so that's the next bastion. But I think, we obviously are all starting to think about that already as we better understand these mechanisms we're looking to interact with.
Great. Christina, would you like to?
I totally agree with Lawrence. I was also thinking earlier we should highlight kind of the use of combinations, and I think we'll get there. I think that's a very positive and important point to emphasize. I mean, look, I think we've made the point pretty clear here today. I'm really excited about getting to more personalized approaches for patients and learning from the more specific and defined patient populations, being able to expand to broader sets of neurodegenerative disease patients. And I think we're seeing that live and in action, and we're just going to see that more. I'm also just, I guess, excited that I hope we will start to be taking more risks in this space, and I think good risk. I think there's great biology coming out. I think understanding how we can intervene sooner in these diseases is also incredibly important. I think we're going to start to see that more. So I'm really excited about what's to come.
I'm really happy that you brought it up, Christina, because I was wondering what to say next, because everything has been said and very thoughtfully. But I think the last thing you said is actually dear to my heart. We need to treat these diseases as real fatal diseases as we did in oncology. So we need to take risks. And there has been this tendency that everything you do for patients with Alzheimer's or any other thing, they can't have any side effect. But if we keep on doing that, we will never achieve anything. You have to take risk and you have to treat, a little bit aggressively. Why not? I mean, and because if you don't do anything, they will ultimately die. So we have to put this also, and the patients are asking us to do this. We were at the World Dementia Council. There was a patient in front at the first row. And at the end, his wife said, well, I've heard this all day. It's all brilliant. You're doing such a good job, but it doesn't help my husband today. And so they have to voice this. We have to work even harder. And yes, we can take risks because they will take risks also.
Great, thank you. This has been great. Thank you for the discussion. This has been a great conversation. I wish we could continue. I know that we have a lot to cover still, but look forward to maybe revisiting looking at and seeing how far we've come in our next conversation.
April is Parkinson's Awareness Month. This year, we are highlighting Parkinson's disease to exemplify the need for action and innovation on the front line to tackle neurological disorders head-on. Please welcome Todd Scherer, EVP Research Strategy, the Michael J. Fox Foundation for Parkinson's Research, and Andrew Sidoroff, Herdig Stern Professor in Neurology, University of Pennsylvania Perelman School of Medicine.
So Todd, I wanted to tell you about a woman I saw in my clinic the other day that reminded me a lot of the things that have been on our minds as we've worked on PPMI together. So she was a 55-year-old woman with really typical Parkinson's disease and very mild, really. And she wasn't having much disability. But the thing that was really on her mind was not how she is now, but how's she going to be in 5 years and in 10 years, and what the prognosis is for her, and what can we do to improve her prognosis. And this is such a common theme that I hear in my clinic. And it really gets to the issues of like, how can we make a good diagnosis early, and what progress is there on in identifying new therapeutics that might slow down disease progression. And I'm sure it's on your mind too, and on the mind of the foundation. I was wondering, like, I deal with this in clinic all the time, but how do when you think about it in your role as a funder, how do you picture this patient, and how does your work address these concerns about diagnosis and progression?
Yeah, I think one of the biggest challenges that we're facing now is how variable Parkinson's can be, and as you're mentioning, unpredictable. So this is really the hardest thing, and what we're trying to do is develop therapies for people at each stage of the Parkinson's journey. So what can we do for people as they're diagnosed, or even one of the things I do want to talk more with you about is the work we're doing even prior to the diagnosis. And what would help people at that stage, and then as they progress, we have available treatments for early in the disease, but as people reach these different milestones of disability, cognitive problems and posture and gait, what can we do to get better treatments at those stages of disease? We've really started to break down our work across this Parkinson's journey from prior to diagnosis, early diagnosis, and latent disease. And I think what I'm excited about is all the scientific opportunities, but one of the biggest challenges is what you mentioned, is what can we really be learning from the clinical experience to identify the right tools and options for people. So, I mean, one of my great interests right now is what's going on kind of in this period of time right before the diagnosis and into diagnosis. And, how can we identify these people, and what is that going to tell us about their trajectory? And I know this is something that we've been working on a lot in recent years. So,
Yeah, I think that I see more and more patients with risk states who don't have a Parkinson diagnosis at all. I see patients that have family members that have Parkinson's, and they're either worried that that little shake they saw 2 weeks ago that never came back is the tip of the iceberg, or they just want to know, get more information about what is the risk given the fact that they have an affected family member. They hear this all the time. And then the other group of people I see a lot of are patients that have this fairly rare sleep disorder called REM sleep behavior disorder, or RBD, who are at substantially increased risk of Parkinson's. In fact, it probably is a prodromal manifestation of the same pathology that causes Parkinson's, as you know. And I'm fairly excited that for the family members, we have such much better genetic tools than we ever had before, both for genotyping the known genes that for like LRRK2 and GBA. And I think there's an opportunity to find genes in those folks that we haven't found yet. So I think that opportunities, maybe you can talk more about it, like these very large genetics collaboratives have the potential to sort of answer more questions in this regard.
Yeah, well, I'm interested in a couple of ways about the genetics, and I think this has been one of the biggest game changers for Parkinson's in the last decade, is that we have actual, real, tangible targets that we think lead to the disease and can be targeted for therapies, and have a much more smart precision type of approach for developing therapies. For example, like you mentioned, for people who have the LRRK2 mutation, and really correct the underlying biology. So I think there's opportunities today with the genetics for new treatments that could perhaps impact that progression that we mentioned. And as you're mentioning, I think the other thing that's exciting is there's now a number of worldwide efforts to identify new genes, and with a particular emphasis on going to underrepresented populations in Africa and Asia and India, to really get a full mapping of the genetics of Parkinson's. And when I started, I mean, you've probably been in Parkinson's longer than me, but it wasn't believed to have any genetic cause. And to really see how this research and science has evolved to identify specific genes, and now there's lots of groups working on therapies against those genes and mapping the genotype to the phenotype. To me, that's really where the cutting edge has gotten in Parkinson's, and it's a great opportunity. But what's even, I think, somewhat very interesting and confounding is that the clinical features can be different even in people with the same mutations. And I think that's telling us something that we haven't really figured out yet.
Yeah, I agree. I mean, in the clinic, we focus so much on the clinical phenotyping and the clinical heterogeneity. For example, does somebody have, down to a very microscopic level, like do they have tremor in their right hand or tremor in their left hand? And I think we're learning that these kinds of distinctions maybe aren't that useful, but they really don't reflect meaningful differences in molecular pathology or biology. But I think we still hold out a hope that there is a clue in the clinical heterogeneity that would lead to more targeted therapeutics, like along a precision medicine model. And I think that clearly, family history versus no family history is probably a really potentially a meaningful clinical distinction that could lead to appropriate therapeutic targeting. I hold out some hope that the broad phenotype that involves more cognitive problems may reflect different pathology than the phenotype that reflects a pure motor syndrome, the so-called like the Lewy body phenotype versus the pure Parkinson's phenotype. I guess I'm as a clinician, I'm remain optimistic that this clinical specific clinical phenotyping that we do in the office will have some meaning. I also pay a lot of attention now to non-motor features. Both because I think they may reflect distinct phenotypes or subtypes, and also because they're such important sources of disability and determinants of quality of life for patients. So sleep, really important. I think pain is somewhat overlooked. The cognitive features are really important. I think most people would agree that if you could find a way to prevent or relieve some of the disability due to cognitive impairment in Parkinson's disease, that would be a huge win. But also some of the other neuropsychiatric features, depression and psychosis, I think those are there's still unmet need there. Although there's been progress, there's still unmet need, and potentially those are a little easier problems to solve than the challenge of disease progression, which I think is top of mind but very difficult.
Yeah, I mean, I agree completely. I think one of the points you just made is a really important one, is that while Parkinson's has a number of very effective treatments early in disease, they're mostly focused on the motor symptomology, and there's a great need and benefit that could come to the patient population by developing treatments for some of these other symptoms that become quite debilitating, and maybe they're more advanced disease. But the other thing that this highlights, this heterogeneity of disease highlights, that you and I have been working on as well, is how to design better outcome measures for clinical trials that really take into account the holistic impact on patients and not just measuring the motor phenotype or motor symptoms. And it's something the foundation's been doing a lot, which is to get this input from the patient population on what actually is impacting their lives the most. What should we be measuring? What's impacting the functional daily living for these individuals? And it's something we've been spending a lot of time on. To really, as we go to more innovative therapies, we also need to innovate in our clinical trial process. And I know that's something you've been working on, that I've been following your work on in pretty closely.
Yeah. So I couldn't agree with you more. We put patients through a standardized clinical rating scale, and that's the main outcome measure we use in Parkinson trials now. It's called so-called Unified Parkinson's Disease Rating Scale, or the Movement Disorder Society UPDRS, MDS UPDRS. And I'm always amazed. I have patients. I had a patient do it this morning who was in a clinical trial, and they say, "Oh no, not this again." And then they tap with this hand, and they tap with the other hand. And I think the patients are somewhat aware that this is a little bit divorced from real world outcomes that really matter to them. I think it's important because it is sensitive to the UPDRS is important and valuable because it's definitely has led to successful drug approvals as an outcome, especially using the total UPDRS, which includes both a functional scale, the so-called Part 2, and an assessment of motor capacity, the ability to do certain tasks, the Part 3. But it's not the same as do you fall, or do you forget to do routine things, or just a real sense of how Parkinson's affects people's daily lives when they sort of reach the point of needing help to walk around, or needing help to do activities of daily living, or begin to have cognitive problems. I think these milestones that translate into meaningful changes in clinical status are much more meaningful to patients and to clinicians, but they evolve slowly over time. And most Parkinson patients don't really start to hit these milestones until at least 3 to 5 years after diagnosis. And so I think some combination of maybe biomarker tracking coupled with confirmatory confirmation of progression through clinically meaningful milestones might be an attractive paradigm for studying whether disease-modifying drugs for Parkinson's works. I also think that, and you talked about this a little earlier, we also are trying to understand how we can test drugs in the prodromal stages of Parkinson's, really before overt manifestations that would lead somebody to go to the doctor and get diagnosed or visible to patients. And actually, this same basic strategy would work in the prodromal stage, where you could track people using biomarker changes, or maybe very subtle changes on digital sensors, and then confirm the changes over time with clinical milestones like the development of a diagnosis of Parkinson's disease or development of more clear-cut features. So I think that I think this is a very nice notion. I think the challenge would be to make it efficient and a timescale that drug companies want, so they can test drugs and get them to market faster.
Yeah, I'm very encouraged by some of the recent work, though, with with the biomarkers. We've made great progress with neuroimaging, imaging the dopamine system, particularly early in disease. And there's some very exciting work coming out measuring alpha-synuclein seeds as early markers. But I think this is really critical for the prodromal phase because people actually have very little, if any, disability. So we really need to have the right tools to understand how we're having an effect with a potential therapy to prevent future disability. But to me, it's really exciting to have this opportunity in Parkinson's, that we could identify people through their genetics or other biomarkers that are at a high risk for getting Parkinson's, and ultimately perhaps intervene, because that's, one of our main goals would be to prevent Parkinson's from happening. So to me, that's a real excitement as we look into the future of where a lot of the innovation can come in a disease like Parkinson's without, as you're mentioning, without losing sight of the fact that there are patients today that are really suffering from non-motor symptoms and other debilitating illness that we can develop new treatments for. So I see lots of opportunity here in Parkinson's, and very excited by this. One of the things I found really fascinating is some of the recent data looking at smell testing, and what that's telling us perhaps about people at risk for developing Parkinson's. So that's something I've just been really interested in, a very, easy thing to screen people with that might be telling us something about their risk for Parkinson's and other neurodegenerative disease.
Yeah, I've been amazed by how relevant smell testing continues to be for people with Parkinson's disease. People with hyposmia were much more likely to have an abnormal biomarker signature and then develop Parkinson's disease. And in our study, nobody who had normal sense of smell, when we followed them for up to 10 years, got Parkinson's. So having a normal sense of smell seemed to be a sort of really a way of demonstrating that you were at very low risk, and having a high sense of smell was a gateway to further testing, more specific testing, to pick up people who are at risk.
So you can imagine in the future that people would get smell tests maybe at their annual visits, like their physicals, and as a screening tool for maybe future evaluation for potential neurodegenerative disease or other ailments.
Yeah, I mean, it's especially if there was something actionable. I think that if we had a disease-modifying therapy that was, tolerable and could be deployed on a relatively large-scale basis, sort of like population health measures, then I think this would probably be. I mean, it's certainly a lot more efficient than doing molecular imaging on the population. You would start with something simple and scalable and not that expensive, like smell testing. So yeah, I think one vision of the future could be that you would get screened for risk for Parkinson's.
One of the things that I'm always amazed with is, we were talking about how Parkinson's is a slowly progressing disease. And in the PPMI study, some of those participants have now been in for over a decade. And in that course, they're getting 6, 7, 8 spinal taps, molecular imaging. That's the only way we have this data on whether people are hitting these milestones or not. But it's a great evidence to what you're saying of people's dedication to really trying to solve this disease. But I think the open access and the return of data helps to keep the participants involved and engaged. Since we're learning, we're not just waiting to wrap up the study at the end for the learnings. We're learning along the way, and we've made adjustments, added new biomarker measures, added new populations as we learn more about the genetics. So I think it's, in my opinion, it's great emphasis of collaboration and how that really helps advance a field, because this study includes academia, there's industry involvement, government, and it really takes this type of effort to solve a complicated disease like Parkinson's. And I think we're making incredible progress with the understanding of the disease. And I actually wanted to just talk a little bit about the excitement with some of the new therapeutics that are coming on, that are really trying to target the underlying disease process, a lot, particularly around this protein alpha-synuclein, which has been linked to Parkinson's, and to see a number of trials that have made it to phase 2 testing, to me is quite encouraging, that we're seeing some of this understanding now converting into potential therapies for patients. I don't know what your general view is on the pipeline, but I think Parkinson's is on the upswing here for innovation with what we're seeing in the pipeline.
I think we were sort of shooting in the dark at some of the targets in the past, and they turned out to not be such good targets. But I think I'm quite confident that synuclein is an important target, and that reducing synuclein aggregates is an important therapeutic avenue for Parkinson's. And I think that it's there's small molecule approaches, there's passive and active immunotherapy approaches, and I think that I'm really excited about this line of research. I think it's just so logical when you think about the pathology of Parkinson's, and I don't think it'll happen be perfect on the first shot. And I think that people, they'll have an idea that this is a concept that might work, but I think it will hopefully we'll see some proof of concept in this early round of anti-synuclein therapies, and then the drugs will get more potent, more selective, and safer, and over time. So I think this is a really important area, but I think we'll still need to be patient, because I don't think we'll get it right the first time. But I think I don't think we should give up if it's not perfect the first time. And then the other area I'm really excited about is the genetically, the targets that are identified through genetics. The obvious ones, the GBA and LRRK2 targeted, but also the ones that come from larger screens that suggest pathways that may be affected. And what do you think?
Yeah, I was going to talk just a second about the pathways, because I think that's interesting, because there's also overlap with some of the other neurodegenerative diseases like neuroinflammation or protein degradation. And I think there's a lot of opportunities there to develop therapies that could perhaps be applied across neurodegenerative diseases, and really gets to the importance of patient selection. So having the biomarkers to select the right individuals to test these therapies in. But I think we're seeing a lot in neuroinflammation as being a really promising area. Mitochondrial dysfunction, also a lot of activity. So it's a quite robust pipeline. We're improving our preclinical testing, but I think ultimately it will be important to do these biomarker-based clinical trials to really move these forward. And I think that's where kind of bringing the clinical experience with the basic science together will be a great opportunity.
Yeah.
The thing that I think is exciting is also just some of the new modalities like gene therapy and cell-based therapy, and some of the new neurostimulation paradigms also have great potential, I think, for Parkinson's disease.
Yeah, I think it's really important. And we talk a lot about molecular pathology and stopping neurodegeneration, but I agree with, I think that some of the therapies that are related to changing, misfunctioning circuits to produce immediate symptomatic relief are really important. I've been amazed by how many patients are interested in the focused ultrasound therapy. And the thing that's really remarkable about it, the ultrasound is interesting, but the thing that really makes it work is the real-time MRI. And so it's the blending of these two technologies that basically takes the old-fashioned pallidotomy and just makes it super accurate. I think patients with established Parkinson's who have bothersome symptoms, either bothersome off time or bothersome tremor, it's a great, these innovations are going to be very meaningful for them while we're waiting to see how the neuroprotection and disease modifying therapies pan out.
Yeah, one of the other applications of technology, I think you might have referred to this before, is just on understanding and tracking of the symptoms. So Parkinson's as a motor disorder does lend itself to some of the wearable tracking technologies. And we've been exploring some of this in PPMI. And at this point, it's still, I think, in more of the experimental testing. But that would also, I think, be a very significant tool, not only for clinical trials, but also for the management of patients. If the doctor could be getting more real life feedback on how the patient is doing, So I think there's just a lot of opportunity for technology to be moved into the Parkinson's space, given what we know about the disease today and what we can still learn.
Absolutely. I think even like passive monitoring for the prodromal phase has potential applications. I think we have to see where this technology leads, and it hasn't really borne its full promise yet. I think we've been maybe a little bit frustrated that it hasn't gone faster, but I think there's probably broad consensus among Parkinson doctors that this is going to work, and it's just a matter of time, and it's going to be an important part of what we do. And I would love to know what my patients are like at home. It's actually sort of an interesting thing. Because of COVID, I do kind of know what my patients are like at home. I do a lot of telemedicine, and actually, this is another thing that's probably really important that in terms of like practice innovation. It's hard to imagine there's not going to be more telemedicine than there is now. It's going to be sort of here to stay. But you see people in their houses, and they look so different than they do in the office. They're frankly generally much better. I find that they look like they're much more comfortable and functional in their houses than they are in the office. So I think that being able to get a sense of what people are like at home is important for research, important for clinic, and we haven't talked much about like practice innovation, but I think that in the last couple of years, definitely telemedicine is the elephant in the room as far as like clinical practice.
So if we started by talking about the patient you had just seen. If we wanted to look in the future, like 5 or 10 years from now, do you anticipate anything being different in that interaction, or what do you think would be different in terms of what you could offer the patient, assuming that a lot of what we said comes to fruition?
I think that biomarker-based diagnosis or biomarker-assisted diagnosis is right around the corner. I think that we've resisted it partly due to sort of the tradition that Parkinson's is a clinical diagnosis, and partly because the technology we have now is expensive and a little bit hard to access, partly because it's expensive. But I think that like these seed assays, and maybe combined with like something simple like a smell test, are not expensive, and they're very scalable. And I think we'll probably use them more, and we'll use them make a more accurate diagnosis earlier. And then the next question is like, how can we make that early diagnosis important? And I think the idea would be to, identify things that can change the long-term course, and that's really going to depend on therapeutics. And I guess I'm optimistic that the pathways that you mentioned, inflammation, for example, and certainly synuclein and the synuclein propagation, I think is really important. And genetics, I think, tailored precision medicine approaches for people with known genetic mutations and understanding more like the polygenic sort of the contribution of patterns of genes rather than one specific gene is going to be important. So I think precision medicine based on multiple genetics plus other things is going to be an important part of prognosis and hopefully therapeutics also. And I've never been more optimistic, actually, that these things are really possible.
Yeah. I share your enthusiasm. I think we're on a chance here for some watershed discoveries in Parkinson's, and we're really establishing the infrastructure and the process for testing them. So I think the future is bright, but we really need to keep the investment going and encouraging the collaboration, and also getting as many people involved in pushing in this field as possible to reach that potential outcome.
Yep.
In tackling cardiovascular diseases, what have been the major hurdles for innovation and investment? What would it take to accelerate product development? How are new modalities and new technologies changing the game? Please welcome Sekar Kathiresan, CEO, Verve Therapeutics, Michael Mendelsohn, founder and chairman, Cardrion Pharmaceuticals, Christopher D'Souza, director, Broadview Ventures, Claudio Ulrich, CEO and co-founder, Cardior Pharmaceuticals, and Simone Fishburne, VP and Editor-in-Chief of BioCentury Inc.
Welcome to the WuXi Healthcare Forum 2022, where we are going to discuss innovation on the front line as it relates to cardiovascular disease and get into some of the issues facing cardiovascular disease drug development. I'm going to launch straight in. I think that we know that there is a dire need in cardiovascular disease for more treatments, better treatments, and more investment. We'll get into all of that. But Michael, I want to start with you, and let's take a longer term look. Before we think of the grim statistics of today, perhaps you can talk a little bit about the successes that have been in the field and outline some of the big changes that maybe we have come to take for granted.
Thank you, Simone, and I'm delighted to be here with my esteemed colleagues, Claudia and Sekar and Christopher. It's an important topic. There was a major decline in deaths from cardiovascular disease achieved from 1960 to 10 years ago. And then an amazing set of advances in the last 10 years. You know, we first developed coronary angiography in about 1960. And we identified key risk factors for coronary diseases like elevated blood pressure and elevated cholesterol. And then several techniques and medicines evolved. We had the development of the first HMG-CoA reductase inhibitor, or statin, in the mid-1970s. We had coronary angioplasty first developed by Grunsing at the end of that decade. And then over the next decade, we identified multiple approaches to thrombolysis in the setting of coronary disease and began to turn attention to heart failure with the initial identification of angiotensin converting enzyme inhibitors in the SAVE trial back in 1992. And throughout this time, there was evolution of using percutaneous coronary interventions, and drug-eluting stents were developed after bare metal stents. So we had a dramatic decrease up until about 10 years ago in coronary disease. And then we've had further advances. So the treatment of coronary disease, of course, has as its underpinning atherosclerosis. Emerging cholesterol therapies, such as the first generation PCSK9 inhibitors, validated the mechanism of that genetic target. And long-acting synthetic small interfering RNA against PCSK9, inclisiran, is now on the market, which works to reduce circulating levels of LDL cholesterol. We've had dramatic advances in heart failure management in the last decade. 4 classes of drug now for the treatment of heart failure with reduced ejection fraction. And for the first time ever, we can treat heart failure with preserved ejection fraction with 2 different approaches. And I think it's important also to mention the dramatic evolution of devices and percutaneous procedures, including, as I mentioned, the development of the percutaneous coronary interventions, both angioplasty and stents, but also implantable defibrillators and transcatheter approaches to valve replacement and catheter ablation for atrial fibrillation. So there have been enormous advances, and yet, as we will, I'm sure, discuss, there is a great deal of residual unmet need. Thank you, Simone.
Thanks, Michael. So, Chris, I'm going to turn to you. It's very clear that there is a lot of innovation. We hear, you know, even in the last few years, and some of the things Michael talked about, there's new targets, even outside of the activity in the device world, which is, as you point out, is very robust. So, you know, you're an investor in this space, and there aren't a lot of you, to be honest. But what do you see perhaps as the major structural hurdles at play, and what are the big needs as you look at it from an investment standpoint?
Yeah, thanks, Simone. So as you said, in the medtech space, which is what we also invest in, since we cover all of cardiovascular at WuXi Ventures, there is a lot of involvement from the acquirers, Medtronic, Boston Scientific, and the such, who grow their business by acquiring technologies. And most of these medtech companies are, single asset companies. And many of these strategics are already involved through investments in these companies. So they are in the ecosystem playing from a very, very early stage, and they want to see clinical proof of concept, which is a lot less expensive to get to in the medtech space than it is in the drug space. In the drug space, traditionally this has been approvals through outcome studies. So we have to find ways in which we can move these programs forward. And when we make an investment, we have to make an investment on the premise that if we don't get an acquisition post clinical proof of concept, then we're going to have to take it all the way through. And so we've got to find ways in which we can take these large diseases and fractionate them down into smaller cohorts. A good example is orphan indications, or smaller genomic-based indications where we can then take them forward. I think also, as oncology has done so well, that perhaps is a much better way to get to the bigger markets by taking these in small bite size. Once you have an approval, it's a lot easier to expand your market through phase 4 trials than it is to get it through phase 2, 3, and to an NDA. So what entrepreneurs have to look at is, if they're going to come to the investment community, come with programs that sort of have the smaller investment requirement to get to market.
Thanks very much. So I'm going to turn to Claudia now. You're an innovator. You have had to forge a path through all of this, which isn't easy, perhaps. Maybe you can talk about what you're optimistic about and how that journey has gone and is going for you.
Yeah, thank you so much. And as Chris pointed out, it's always difficult for smaller companies to develop and discover transformative treatments for patients in the cardiovascular space in the larger market. But on the other hand, and there is the optimism, that we are confident to move towards further personalized treatments, as at the final end, in the center should be the patient and the patient's well-being. And in that regard, the oncology field, I would say, has paved the way with that approach. And we at Cardio, and I think also my other colleagues like Mike and Sig, we are committed to developing specific and curative approaches for cardiovascular patients to improve finally the quality of life long term. And I would say in the last couple of years, the use of AI and big data and other computing technologies have become even more important, especially during the pandemic. They have improved numerous processes which are involved in early development, such as target selection, drug optimization, as well as in later stages of development, including conducting efficient clinical trials. We at Cardio, we leverage these technologies to target specific patient populations more efficiently and to optimize and improve our clinical trials, to accelerate discovery and development processes, and using also bioinformatic models to improve drug development, specifically in the non-coding RNA space, which is clearly a next generation target.
We're going to return to some of these themes later in this conversation, but I want to go to Sek. Yours, I would say you've kind of got an edgy technology with base editing and in a difficult field. As you look at what you're doing and what others are doing in the field, do you feel that we're going to enter an era of just incremental improvements, which there's nothing wrong with incremental improvements, but is that where we're headed, or is there really an opportunity in cardiovascular disease still to deliver something transformational?
Thank you, Simone. Thanks for the opportunity to participate. And no, I think there are opportunities to really be transformational in this field. And Verve Therapeutics, the company that I lead, is looking to take on one of those transformations. And specifically, I think one of the challenges with the way we manage a chronic disease like atherosclerotic cardiovascular disease, like heart attack, is we manage it with chronic care. So daily pills, intermittent injections, multiple pills a day. And that model is broken. That model being broken is the reason we have such high prevalence, one of the reasons we have such high prevalence, such a strong disease burden still, despite what's available. Because there's a simple stat. If you take 100 patients who have a heart attack today and ask a year out from their heart attack, how many of them are still taking their cholesterol-lowering medications, which is the #1 way to make sure you don't have another heart attack. And Simone, it's less than 50% are still taking their cholesterol-lowering medications. So what we're looking to do at Verve, for example, is to transform the care from chronic care model to a one and done. So think about it as a one-time treatment, permanent lowering of LDL cholesterol. If this can be done and be safe and effective, this is a key answer to heart attack. And so I think there are opportunities to be transformational in this field. And I think what we're trying to do, which is gene editing, to have these one and done treatments, is hopefully one of those.
I'm going to go back to Michael now, because you gave us a very elegant overview of all the good news, of all the success stories. Maybe you can respond to a couple of the things you've heard. Talk a little bit about what you think are the key needs, and maybe do you agree that there's a lot cardiovascular disease can learn from oncology, and if so, what would that be?
Well, I think I do agree with that, and I think one of the key learnings is related to some of the key hurdles in the field that were just discussed. There has been, thanks to oncology, a clear focus on what is called precision medicine. And one of the key hurdles in cardiovascular drug development is trying to design trials that are thoughtful and that are focused in a manner that Christopher alluded to. We are in some ways a victim of our own success, because all the new therapies that are needed must show a benefit when they're added on top of standard of care. And this has the propensity to lead to larger, expensive phase 3 trials. And it's key to understand how to best translate phase 2 findings to phase 3 trials. And at the core of that, we need to focus on what I think of as phenotypic refinement and novel biomarkers. So we need to understand subsets that we can study and achieve proof of concept within, and use novel biomarkers or evolved biomarkers to inform more thoughtful study design and do a more focused and tailored approach. Clinical development plans that avoid the cardiovascular mega trials of the last decade and instead are able to obtain proof of concept in a way that is focused and well-designed, emphasizing phenotypic refinement, looking to use strong biomarkers and adaptive designs in the trial design, I think are where we are and where we should continue to evolve.
Maybe I can throw this to the whole panel. So what needs to happen to make these genetic approaches more broadly available? Is there, is it going to be dependent on changes in clinical practice, like having routine genetic profiling? Are there other systemic changes? How do we go from sort of people understanding it on this panel to it actually happening?
And may I start? So I would say with the American Heart Association that issued in 2020 a statement that the genetic testing for patients diagnosed with all forms of cardiomyopathies and antiarrhythmic and lipid disorders, I think they had included a list of 30 medically actionable genes. So that's one thing where we started through. But the use of cardiogenomics finally also needs to end up in results. So as we all try to improve the treatments in that field, we have also to make sure that we improve the knowledge with practitioners that are involved also in clinical trials and treating patients, So on how cardiogenomics can improve treatment and disease outcomes. So these are the people who will collect data and screen large patient population for the markers. So they also have to be involved and have to be trained. And I think also the regulators need also to be involved. But as to my experience and knowledge at Cardio, we have made good experience with the FDA, also working on several surrogate endpoints in larger clinical trials. And as Mike said, coming from proof of concept to pivotal trials without only having 10,000 to 15,000 patients involved. And I think there is big movement in the whole scene.
And if I can just, I totally agree with Claudia, and I was going to point out to the fact that right now, the standard of care is a patient comes in, they put on one drug at a low dose, then you go to a next high dose, and you add something. So it's really efficacy by trial and error. There is no logic to it. And yet there is a growing body of evidence to show that different patients respond differently to certain types of drugs. And so why are we not, including that information in our prescription procedures and how we treat patients? So right now it's all patients get the same drugs. You start with one combination, keep on adding to it, when we know all the patients are not the same. And so we've got to start bringing into or changing the way we treat patients and changing standard of care to incorporate genomics and proteomics and these sort of features into optimizing right from the very beginning and getting away from this trial and error method.
Yes, Simone, if I could add, I think what Mike was talking about is, having a well-defined patient population and getting to human proof of concept as quickly as possible with appropriate biomarkers that will translate into clinical benefit, right? So if you think about all those criteria and people are looking for the right patients, the right biomarkers for all the different subsets of cardiovascular disease, for atherosclerotic cardiovascular disease, the poster child of this all working is actually LDL. If you think about, for example, our phase 1 study is going to be in 40 patients. These are patients with a genetic form of high LDL, heterozygous FH. LDL is the approvable biomarker, genetically defined. Now, we're not testing a new mechanism. We're going to be testing PCSK9 inactivation, but we're testing a new modality, basically a one-time gene edit in the liver to turn off the gene permanently. But we'll be able to get that proof of concept in about 40 patients. And so I think to the extent that we can really get this kind of model translated to other areas of cardiovascular disease, for example, heart failure, which I think Claudia is working on, or rhythm disturbances, or even some of the other more complex, like pulmonary hypertension, for example, is a great example where I think this is going to work, where genetically defined, you have some treatments, you have an endpoint like a cardiopulmonary exercise test or a 6-minute walk test that is approvable. So there's progress, I think.
So, Sek raised the issue of new modalities, right? So you might be going in with the gene therapy, and several of you have talked still about things like LDL, targets that actually are not new targets. We know a lot about these targets, and they still seem to be areas related to a lot of the high unmet need. So what is the role that new modalities are going to play? One thing I want to add is that we tend to bucket new modalities into this phrase, but there's RNAi, there's gene therapies, there's cell therapies, there's ADCs and bispecifics, sort of a host of new modalities. So maybe the panel can talk about generally whether we're going to enter an era where new modalities are difference makers, and which are the ones that you think will actually sort of forge ahead. Go ahead, Michael.
Thank you. So I think the first thing I'd like to say is at Cardurian Pharmaceuticals, where we have 3 programs at present and are developing additional, we believe it's important to be agnostic to modality. And it's about understanding the targets and the pathways well. And target identification and validation is most powerfully achieved in two ways. The first is genetics. And Sek just gave the example of LDL, which of course was a genetically validated target from the seminal work of Brown and Goldstein 3 decades ago now. And it's evolving further, but it was identified beautifully and using genetic approaches. And then the second way is clinical validation. So, for example, the natriuretic peptide receptor pathway is now clearly clinically validated by the enhanced and increased efficacy provided by neprilysin inhibition in the setting of that combination with an angiotensin receptor blocker. So I think it's important to be agnostic to modality and to think about the best ways to drug the targets and pathways that are validated. And the new modalities that we're now beginning to discuss are exciting in that regard. You know, we have 2 companies represented here in Sek's and Claudia's that are advancing novel and new modalities to treat major cardiovascular disease. And there are areas that are still, after many years, not quite ready, such as cell therapy. That's still being explored. And we don't really have a shining example at present of a cardiovascular cell therapy. But we do have examples of cardiovascular gene therapy. And to Christopher's point, we're advancing those especially in smaller or rarer diseases where we can get proof of concept and powerful intervention in some very difficult to treat diseases.
If I may add something, because I think, Mark, you're totally right. And also Chris as an investor is engaged in a company developing also, like we do, the non-coding RNA space, which is clearly a new modality. On the other hand, has matured over the last couple of years. And yeah, we are building really on deep root of RNA biology. And if you know that these non-coding RNAs amount to the vast majority of the RNAs, and only, I would say, roughly 5% are for coding RNAs representatives that do proteins, I think that is a really untapped treatment modality. And we try to pioneer, on the one hand, this approach. On the other hand, we also do have really good data, and that's something we are all looking for in cardiovascular, to have the right animal models for developing such kind of new modalities and to show clearly efficacy and translatability from the cell culture dish to small animals and large animals, and finally to patients.
This has sort of been an extension of how biology has changed too right now, our understanding of biology. I mean, 30 years ago it was all about small molecules, and then came the biologics as we got to understand better the role of antibodies. And now we're finding genomic targets that are not small molecule druggable targets. And also in parallel, the whole technology in the space of ASOs and gene therapy and CRISPR, et cetera, is moving in parallel and allowing us to, which 20 years ago we wouldn't be able to do, but allowing us to now drug these targets that are not amenable to small molecule or antibody technologies, right? And so it's kind of both of these are running in parallel, understanding of the targets as well as technologies that are now making it possible to target these new targets that typically would not be antibody or small molecule targetable.
Yeah, I think Simone, if you asked like, what role do new modalities have to solve this problem? I mean, the new modality has to solve an unmet need, and so if you think about kind of what we're trying to do, it's built off a very simple insight that goes back now 30, 40 years, is that if one's LDL is low, lifelong, it's really hard to get a heart attack. And then the question is, how do you get somebody's LDL low? I'm talking about 30, 40 milligram deciliter lifelong. Because there are people walking around who naturally have it that low lifelong. And the reason is they have specific genes switched off, a cholesterol raising gene switched off. So we wondered, could we actually do that through a medicine and get one of these cholesterol raising genes switched off, mimic basically the natural resistance mutation situation. And then the technology came along, CRISPR base editing, that allows you to precisely inactivate and turn off, just change one letter in the genome, basically surgery without a scalpel, molecular surgery in the liver. And if this works and it's safe, then it'll meet that unmet need of being able to get somebody's LDL very low and lifelong low. So I think in the end of the day, the new technology has to solve an unmet need.
So if I may make a response, I think that was beautifully said and summarized. And we know there are many new modalities: gene editing, gene therapy. We have the biologics now for quite some time, novel antibody drug conjugates, degraders like Protacs, the mRNA therapies. It all starts, as Sek pointed out, with the unmet need. And I think it's important, in talking as we did originally about reasons for innovators to have optimism, to remember that there's huge unmet need in cardiovascular still. Atherosclerosis is the example we've been discussing, clearly a major remaining unmet need. Heart failure, especially HFpEF. The arrhythmia diseases, especially atrial fibrillation, and a number of inherited diseases like the channelopathies. And pulmonary arterial hypertension, which I believe Sek mentioned earlier, still it's a rare disease, but a devastating disease. There's also a need for peripheral vascular disease therapies. And there's a need for next generation antithrombotic therapies, where we attenuate thrombosis without disrupting hemostasis. We have the factor 10 inhibitors that have been very successful, but they're going to need to be improved on. And we're doing that with, for example, factor 11 inhibitors. So there's this huge number of unmet needs still in cardiovascular that we can approach. And it's very exciting to imagine some of the newer modalities being brought to bear in the manner that Sek just described for gene editing.
So I'm going to follow up on that, thank you, with turning the conversation to sort of critical issues in innovation, critical scientific or technology hurdles that, if you solve them, could accelerate many therapies. And I want to go to Claudia to start with, but everybody should weigh in. Because Claudia, you talked before and previously when we chatted about the importance of data science, the ability to analyze large data sets and use AI models. And maybe you can expand on how you see that for this field in particular.
Yeah, as I said, I think employing the diagnostics, including also proteomics, artificial intelligence, and imaging, that can characterize the status of a cardiovascular disease patient, and can give you more information about the disease burden and the localization. So, and this important process, I would say, can really become the driver of a shift from systemic medication towards targeted and tailored therapy. And the scale requirements, as pointed out earlier, of clinical trials that puts both a financial and a time burden to our relatively smaller biotech companies, getting access to dilutive and non-dilutive funding. And that will remain, in my point of view, an incredibly importance in the field.
So until now, the panel's been in broad agreement about pretty much everything. Is there agreement on the need for better data science and even AI models? And how sophisticated do people think that is?
iWatches do a lot more than the devices that are in the hospitals, where they come in every 2 hours, take a blood sample, take your blood pressure, when really they should just have a patch on you that sends out the signal and leave the patient alone, right? I mean, that is the level of technology that we have in our hospitals today. And, there's a lot of room for improvement of that. I think also, while we talk a lot about having new drugs, and there is a need for new drugs. I mean, to put things in perspective, the last drug for AFib that was approved was in 2009, Neurodegenerative. There's been nothing in that space because they require large trials and safety has been an issue. But let me remind people in the audience of here who are entrepreneurs, innovation can go across multiple areas. I mean, how do we better use the drugs that we already have, right? We have a lot of drugs in the basket for physicians. How do we improve the use of those drugs? How do we reduce compliance? SAIC is doing that in one way. How do we decrease bill burden, which most of these patients are on 4 or 5 drugs? But on the other hand, we've still got heart failure with preserved ejection fraction, which is looked upon as a single disease when perhaps it really is a syndrome. How do we break that down? How does genomics help us break that down and find cohorts of there where we can start to run clinical trials? So there's innovation across the spectrum from what we already have and using it better to what we don't have and finding ways to get to treatments.
I think one item I'd like to point out, Simone, in terms of areas of need is delivery. And particularly for kind of genetic medicines, our drug is delivered to the liver, our gene editing drug, because that's where cholesterol synthesis happens. That's where the relevant gene is. But it'd be nice to have a way to deliver things precisely to the myocardium. That's been a big challenge for the field, both from cell therapies, gene therapies, gene editing medicines. There's just not a good delivery vehicle. And the people who solve that, actually, there's a lot that a benefit that could come from solving that challenge.
Michael, did you want to add to that?
It's a very important point. I wanted to make a more general comment about our innovating with novel technologies. The cornerstone for all of us is safety.
Yeah.
And ensuring safety is critical. And the innovative technologies are often associated with less understanding of potential safety concerns. And small molecules have an advantage in that regard because we have so much more experience with them in drug development. But the critical attention to safety as we evolve the new therapies is going to be very important and fundamental. And I admire greatly, for example, what Verve has done in testing their approach in non-human primates. It's the second best animal model after humans. And I think all of us think a lot and regularly about safety concerns. And so the bar for safety is higher for newer modalities. But there's enormous work going on, and I'm optimistic that we will carefully evaluate the safety of various approaches. I think no novel modality has been more challenged by this than gene therapy, as most of you listening to this panel know. So I just wanted to make the safety point. Thank you.
Yeah, and I think along that line, one more comment along that line, Simone, is that the things evolved very quickly. I mean, I think it wasn't that long ago, I'm sure, in the early 80s, Mike, that people thought the monoclonal antibodies were very scary.
Right.
And then remember, in the 90s and 2000s, people thought RNA-based therapies, how could you get that to work? I mean, siRNAs might have all kinds of off-target effects. You might be suppressing other really important RNAs. What about the specificity? And then now you have an siRNA approved for cholesterol lowering, a market for 20, 30 million people. So I think that, and that's all happened because of what Mike mentioned, which is people show safety in large numbers of patients and then get the ability to move forward into an approval. So I think it'll all be, the proof will be in the pudding, and I think the onus will be on us to to demonstrate that in a, initially in a high risk group, and then ultimately, hopefully in a broader population as well.
But if I may, I'd like to add, because I think this is very important to make that statement also on safety. But as you said, the proof of the pudding is clearly, phase 2. I've seen and experienced that there is also a change in these animal models and also toxicology investigations. So what we did before starting our clinical programs, we were also with the FDA, and they clearly advised to also look into larger animals like pigs. We did a 2 species chronic tox testing, for example, in rodents and pigs. And we had also one of the largest pig trials investigated in a post-myocardial infarction model, as well as in a chronic heart failure model. And I have at least the feeling that the landscape is a little bit changing. So there might be for some of the new treatment modalities still the non-human primate model, the best one and the most safe one, but also the pig trials and the pig tox investigations have come to speed and showed clearly good results, mimicking also the translation into the human data.
What role do people see for public sector organizations, NIH, not-for-profits, academic organizations, to maybe accelerate the field either by developing animal models or going to what you talked about before, I think, Sate, which was solving delivery challenges for some of the new modalities? Is that something that you see a role for academic organizations to do, or academic investigators, or nonprofits, or is that going to stay in industry? Is that where we're going to see the advances being made?
Yeah, I was just going to make that point, Simone, that, for example, in heart failure, there HFpEF, we don't really have a very good model, and it's probably going to be multiple models depending upon what aspect of HFpEF you're dealing with, if you consider that disease to be more of a syndrome. And I think that's really where NIH funding and academic collaborations, will help.
Yes, Simone, I think my own experience trying to develop this new medicine, new modality for with gene editing, I think may give people a sense of the balance of what academia can accomplish and what is needed, in the private sector. So, when we started in 2016 with this idea of a base editing medicine, the components of which are mRNA, guide RNA, package, and a lipid nanoparticle. Let's think through kind of where those all came from. Almost all of those components come originally, they're inventions from academia. So the mRNA, everybody's familiar with now based on COVID vaccines. The base editor came out of David Liu's lab at Harvard. The guide RNA technology kind of also out of that lab. The lipid nanoparticle comes out of University of British Columbia. There's a famous lab there that did a lot of that early work. So all of the initial observations kind of are seeded by academic labs, often with NIH and public funding. But what was clear to me back in 16, 18, when we're developing this idea, that to take it to the next step and actually make a medicine, it was not going to be possible just, So, we've raised $500 million over the last 4 years to develop this medicine. That's like, I don't know, like 1,000 R01s. There's just no way that you could do this in academia. And so, so there's a role, there's an important role for each. And I think people need to recognize that and value and cherish the respective contributions.
So I think we have time for one more question, and I want to go to the other end of the drug development spectrum, maybe not exactly the other end, it's throughout, but to ask the panel finally whether you think there are regulatory changes that are really needed to accelerate product development in this field. New endpoints you referred to before, Claudia, or anything else. Chris, you're smiling. I'm gonna go to you with that one, and then ask the rest of the panel.
Oh God, I'm a scientist by training. By the time things get to regulatory, I'm that I'm on my 4th or 5th different program. But I do think that there have been changes. For troponin, for example, is a good biomarker that the FDA will accept as a surrogate for outcomes for heart attacks. But I think we need other surrogates to be provided, especially in the heart failure space, right, where we're not going to be able to do outcome studies. Atherosclerosis is another one. We're caught in this conundrum, how do you get a drug approved for atherosclerosis? You're not going to do an outcome study, but at the same time, imaging is not a regulatory endpoint, although that's pretty good. So I think we're going to have to work with the FDA and vice versa to come up with some earlier markers that could allow for the approval with then, some increased phase 4 pharmacovigilance.
In our interaction so far with the FDA for this new modality, they've been very, very knowledgeable and kind of on top of the detail. So they actually put out a guidance a few weeks ago on what they're looking for human genome editing, particularly in vivo liver editing. And I think having that kind of standards and publicly pronounced standards is actually a great move for the field. And so that's the kind of thing I think I applaud them for that, and they need to continue to do more of that, because when there's uncertainty is when there's people don't there's a reluctance to invest. But when there's regulatory certainty, whatever the bar is, as long as you know what you need to do, people like that because they know the bar they have to cross. So that document, for example, is a good example of telling the world what the bar is going to be from their perspective.
I think the speed in regulatory approval procedures could be enhanced, and maybe benchmarking the approvals of the mRNA vaccines, for example, we could also think about some rolling submission. We would need more in surrogate parameters like biomarkers, for example. So new biomarkers, maybe also surrogate endpoints that translate from phase 2 into phase 3. And would be accepted, and putting also more the quality of life of patients in the center. So that would be, if I could say or phrase a wish to the regulators, I would phrase these two.
Michael, last word to you.
Well, thank you very much. So we're developing 3 programs at present at Cardurian: a heart failure program, and a arrhythmia disease program for a rare arrhythmia disease, and an antifibrotic. And in our interactions in the first 2 with the agency, I've been impressed at their commitment to innovation. Their job is to make sure we do this safely, but they have been very open to discussing novel approaches. And I believe they care, as we all do, about getting to the place where we can develop these newer modalities, and they can be agnostic to modality. I'm also encouraged that our new FDA commissioner is a cardiologist. I'll leave it with that.
Great.
Well, thank you very much, Michael, Claudia, Sek, Chris, and of course, thank you to WuXi. This has been a great conversation.
Thank you, Simone. Thank you all.
Thank you.
Cancer therapies are transforming patients' lives in ways that were unimaginable 20 years ago. What are the frontline innovators trying to address next? How would advances in cancer biology and new technologies further transform the field? Please welcome Rajesh Chopra, Venture Partner, Head of Oncology, Appletree Partners, Adam Friedman, President, Corporate Strategy and Business Development, Scorpion Therapeutics, Luba Greenwood, CEO, Cogent Therapeutics, KT Mortgott, Healthcare Venture Investor, Investment Partner, Droya Ventures, Lewis Williams, Co-Founder, Chairman and CEO, Walking Fish Therapeutics, Inc., and Dave Madge, VP of Discovery Services, WuXi AppTech.
Hi, everyone. Welcome to this panel discussion in which we'll take a look at what the future may bring in terms of next generation cancer therapeutics. I'm very pleased to have a distinguished panel of leaders from oncology discovery and investment teams with me today. I'm looking forward to hearing what the group have to say about recent developments and future trends in oncology drug discovery and what they may hope to be able to deliver to patients over the coming years. I'd like to start by inviting each of our panelists to spend a few minutes each telling us what excites them about their current projects. Perhaps first with our panel members who are driving cutting edge companies, Rusty, Luba, and Adam, and then our two investor panelists, Raj and KT, to learn more about the impact they hope to have by supporting new company formation and development. Rusty, would you like to start? What do you hope the B-cell engineering platform at Walking Fish Therapeutics will deliver?
Sure, thanks, Dave. My current work is at Walking Fish Therapeutics, where I'm a co-founder and CEO. I think that what our field needs is to focus on durable, long-lasting therapeutic benefits that are safe and tolerable. And that requires some risks. And so at Walking Fish, we're doing two things. In oncology, we're using B cells, taking B cells from peripheral blood and engineering them with chimeric antigen receptors, analogous to those of CAR T cells, but different in the specifics. And those are designed to recognize tumor antigens on the surface of tumor cells and to display those antigens on MHC and generate an immune response. In addition to B cells, the CAR B cells carry payloads that further enhance immunity. So the goal here is to get a long-lasting response against cancer cells by using B cells for their very superior antigen presentability. The hurdle in this field has been engineering human B cells, and now we've learned to do that very effectively. The second part of our company, which is not a top topic for this meeting, is to engineer B cells to differentiate to plasma cells and produce proteins systemically. And here we want the B cells to be long-lived, produce proteins that are missing, for example, in monogenic protein deficiency diseases. And here we're going for what I would call near cures, that is very infrequent therapies, maybe every few years. And so both arms of our company are focused on long-lived therapeutic effects. So I'm excited about the field of oncology for many reasons. So I don't want to take too much time here, but I think there's a lot going for oncology now in terms of the future. And I'm very excited about the prospects, and we can go into that once my colleagues have weighed in on this.
I'm sure we will. Thanks, Rusty. And there is a strong sense, I think, from the other panel discussions as part of this forum of, a strong feeling of momentum into new technologies, into new directions that are going to yield big steps forward in the area over the coming years. And we'll come on, I think, to talk interestingly about what those developments will look like over the coming years. But before we get to that, Luba, you're looking at particular cell states in Cogent Therapeutics, right? A little bit about what that means for you.
Absolutely. I'm the CEO of Cogent Therapeutics. We're based here in Boston. I got to know Cogent as the founder and managing partner also of the Dana-Farber Cancer Institute Venture Fund. We started the venture fund for the Dana-Farber because we recognize, and this is important for the discussion here and really for the conference overall, is there are a lot of advancements in oncology, but at the same time, the prevalence of cancer and the cancer therapies that we currently have on the market, while many effective, we still can do a lot more. So the reason behind the Dana-Farber Cancer Institute Venture Fund was to find the true breakthrough therapy. So not another Me Too or another drug that perhaps extends life for a few months, but something that's truly effective and transformational for many cancer patients and for many different cancer types not just for a few cancers. So when I was looking at the various investments and investment opportunities, as I mentioned, there's some exciting technologies, but I don't think enough. When I came across Cogent Therapeutics and learned about ferroptosis, one thing became clear is one of the reasons why many cancer therapies do not currently work is because what we call new platforms and new modalities are all going after the same type of cancer cell death, cell death period, which is called apoptosis. And we know there is resistance that's built up and many therapies just don't work. So when I heard that there is ferroptosis, that there's another innate way that a cell can die, and not just that, it's truly the death can be controlled given what you asked, Dave, in understanding biology and understanding cell states. We can induce ferroptosis and induce this therapeutic type of death and also prevent this type of death. And certainly inducing it in oncology, but also beyond oncology in areas such as inflammation, immunology, but also prevent this type of cell death as well. So ferroptosis, we view, and not just us, but it's wonderful to see in the scientific community, the importance of the different types of modalities like ferroptosis, that we're just at the beginning of this type of an understanding, the cell death.
Fantastic. Again, we will keep coming back to this topic, I think, of technologies, of different strategies and different modalities. So I'm looking forward to hearing what the other panelists have to say on this topic. But again, before we get to that point, Adam, I'd like to hear a little bit about Scorpion Therapeutics. You laid a strategy that you refer to as Precision Oncology 2.0. Can you tell us a little bit about this approach and what you hope to deliver using this strategy, Adam?
Sure, Dave, and thank you for the opportunity to speak with such a great panel on a very important topic. So Scorpion Therapeutics was founded a couple of years ago, led by our academic founders, including Keith Flaherty, a serial entrepreneur and an international leader in the targeted precision medicine space, and also including Gadi Getz, the Broad Institute, and Laurent Barpellet at Mass General Hospital here in Boston. And they founded it with a very clear vision, which was to bring precision oncology to as many cancer patients as possible. I think all of us are aware of the tremendous successes of targeted therapies over the past 20 years. But the reality is that less than 10% of patients actually benefit from targeted therapies. And so the goal of the company was to bring target oncology to more patients. And to do that, you really have to go after more targets that have been pursued before for precision medicine. And then to do that, you actually have to build tremendous capabilities to target these proteins which have never been targeted before. And so what we've done here at Scorpion over the past couple of years is invest significantly in a number of advanced capabilities that fall into three key areas of target biology. We have a tremendous wealth of novel targets from a number of efforts over the past 20 years. We've made investments in chemistry, including medicinal chemistry and the next generation platform for chemical proteomics to address those targets. And then finally, in data sciences across both biological data sciences and chemical data sciences, including structure-based drug design and molecular biophysics, where there's been a tremendous democratization of cloud-based supercomputing power that really allows you to apply these technologies to a much broader group of targets. So Scorpion has integrated these technologies into a single platform and are pursuing more than 15 targets across a range of different categories. And we look forward to our first program entering the clinic in early next year.
Fantastic. Thanks, Adam. Again, we come to this message of more and more diverse targets and technology developments driving the industry forward. I guess, Raj and KT, this must be what you see day in, day out with new opportunities. Can you tell us a little bit about your current activities in setting up and supporting new companies? And perhaps KT, you'd like to start.
Sure. Well, first of all, thanks for including me in this fantastic panel. Really a pleasure to be both with co-investor as well as some very interesting companies. I'm a healthcare venture investor investing in novel therapeutics to treat cancer and genetic disease. And in the oncology side, the focus is on investing in novel therapeutics, approaches that have the potential to transform patient care and improve patient outcomes. This is a grand goal, and I think it's a great reason to wake up in the morning every morning. And I think it's certainly this chance to ultimately impact health in meaningful ways is really what motivates us, a lot of us in this industry. At Droya specifically, which is where I'm an investment partner currently, we're a specialty biotech investor focused on discovery and preclinical stages. And our current portfolio is about 24 companies or so, split between US and EU, with interest in, as I said, really trying to break open new areas in cancer. And among these areas, I think there's a lot of exciting discovery. I think we're hearing about some of that right here in this panel. Discoveries at the forefront of science and the ability to, modulate targets that have been considered undruggable for the longest time, as well as therapeutic approaches that are taking account of not only the mutated proliferative cancer cell, but also the tumor microenvironment. And so those are two areas that I think that we can, that have been brought up, we can talk more about in this call, and certainly are very much of interest to us as investors.
Fantastic, thanks, Katie. Raj, you're building an impressive portfolio of oncology opportunities and companies in ATP. Can you tell us a little bit about how you're going about that and how you find it stimulating?
Yeah, thank you, David, and thank you for inviting me to join this great panel. I suppose we have a model, and we've got the privilege, of being the funding to be sole investors for Series A, B, and often all the way to crossover rounds. And one of the key things that we do, and I know other VCs also do this, but spend weeks and months working with our founders, around novel concepts that can then be channeled therapeutic paradigms and drug discovery programs or platforms. And not only do I work with a really talented bunch of colleagues at ATP to bounce ideas together, distill down priorities, but we all cooperate to operationalize our companies. So, for example, even though I'm head of oncology and a partner at ATP, I'm also chief medical officer of a company out of Boston called Marengo, where we selectively activating T-cell subsets based on the germline repertoire. So that's one area of operationalizing, being part of the founders of that. At the Crick, with a colleague of mine, we've set up a company, the Crick Institute in London, to really see how dendritic cells detect dead cancer cells specifically and then orchestrate the immune system. Really investment, but with really knowledgeable both partners, but also working very closely with founders to really address some of what we think are fundamental questions in cancer biology, and that's what makes this really exciting.
I think this question about, there is still a need to solve some fundamental cancer biology questions to move the science and the basic science forward. We've obviously made huge strides in the last 30 years or so, reducing outcomes for a large proportion of patients. But I think I'm sure we'll all agree that there's much more to do. And 5-year survival rates for some common cancers, like non-small cell lung cancer, pancreatic or glioblastoma, these remain low. So there's also considerable variation in outcomes by geography.
David, I just want to say I completely agree with you. Understanding biology, and this is one of the reasons why, if you look at the interest currently of pharma companies in oncology, they're interested. They're still interested, of course, in companies that are truly platform. But most importantly, it's the biology. So either platform, new modality, or as you say, technology, to uncover more biology so that we actually understand what causes many of these cancers. I think it was a very good point that we're still, I think it's under 10%. I've seen numbers as low as 8% of cancers that we're currently even going after So I think it's very important what companies like Scorpion and Cogent Therapeutics are doing, because they are thinking about biology and they are thinking about beyond just the genomics, and really digging in and understanding what's really causing cancer, so that we can serve more patients.
I agree with that.
I'd like to dig into this in a little bit more. And Rusty, I'll invite you to comment first. I'd really like to try and understand what are the basic science developments that you see on the horizon, that you think are going to have a big impact over the coming years. And Rusty, perhaps you'd like to start.
Sure. I think that this idea of understanding mechanism of oncogenesis and tumor growth is really important. Not only broad mechanisms, but also individual mechanisms in individual patients. And I think the complexity is immense. It's daunting. And I think we have to admit what we don't know, which is what Luba was referring to, and that is we just don't know the mechanism of most cancers. In parallel with that, however, I think it's possible to work on cancers in which you don't know the mechanisms. And that's, I think a lot of immuno-oncology is focused that way. I've been working on this since I worked on IL-2 at Chiron many years ago. And, you know, the body is, in many cases, making an immune response against tumors. It's just not effective in the cases in which tumors actually grow. And so I think there you don't have to understand the mechanism of each cancer. You do have to understand mechanisms in the immune system, though. And so a lot of the basic science that's going on is well worth the investment. And I think that these collaborations between academia and industry, I'm happy to see those proliferate. And I think we need more of those even than we have now.
Would anybody else like to chime in on this topic?
Yeah, sure. I mean, I completely agree, Rusty, with that. I think academia certainly is the lifeblood of our industry, and the continued elaboration of oncogenesis, validation of targets. We depend upon that and certainly use that information to help guide, you know, which targets we should go after. I think in particular, we're now in I think a lull of understanding in immuno-oncology, where there is going to be an immuno-oncology 2.0. And I think a lot of this is dependent upon academia in collaboration with industry to help generate that next list of what are the mechanisms of resistance to the first generation of immune checkpoint inhibitors, for example. And I firmly believe that that's coming, and I think we need that basic science to do that. I think the other area that's really fascinating and beneficial on the sort of intrinsic cell target and small molecule side is the continued advancement, really remarkable advancement in machine learning and AI generated prediction of structures, so AlphaFold, DeepMind-like activities, where it's a real bottleneck for industry. And I think the continued investment there, certainly that's within companies as well as academia, I think that will be hugely beneficial for industry.
David, I just want to make the other point. This is more for policymakers sitting on grants panel in the UK and Europe, is that there's this constant push to talk about translation, etc. But I do think governments need to, and you know, where there's state funding or university funding, we should not ignore basic science. Mm-hmm. That is still the lifeblood, and we continue to sort of feed the innovation that we need in industry and biotech. And in many ways, I think that is where, you know, state funding and university funding, in my opinion, can make a real difference.
Yeah, I would echo that and kind of amplify that. The you know, we can put some numbers around it. You know, for in the U.S., right, the NCI budget in 2019 was about $6 billion. So really a significant portion of that goes to clinical trials, but still it gives you a sense of what some of the research funds are there. And in comparison, we talked about sort of, you know, how much venture investing goes into oncology. So if looking at all of biopharma in 2021 was an all-time high of $36 billion. And based on SVB's data, we're talking about 11 billion in venture capital in oncology specifically. So 2 billion of that is going into seed and Series A. So if you think about sort of all of those funds and sort of what's specifically going into oncology, it makes it a pretty, between the funding and then the innovation that's driving on the academic and company front, I think this makes it a pretty exciting time to be in the translation phase of oncology, right? In this interface between, academic science or early science and discovery. I think that we're seeing, some examples, we've touched on some of these, really looking at new modalities, right? I think one example, I'd be happy to share one example from our portfolio, which is Ambigon, that targets intrinsically disordered proteins. So these are disordered in a way that make them very intractable to small molecule drug discovery. And so rather than trying to bind directly to and inhibit a disordered protein, the idea is to stabilize an interaction with a chaperone protein called 1433. And once bound, then be able to find ways to stabilize that, right, with small molecules and stabilize the interaction. So this and other molecular glue approaches are kind of opening up many new targets. And I think that this is an important, sort of expansion of what we can do here.
I'd like to follow up on that. The molecular glue, I think, is a big advance. If you can localize therapies, then you have a better chance to get a good therapeutic index. Something I've been passionate about for years. Another area where I could see cooperation between academia and industry, I think is truly important, and it's a real bottleneck. And that is, if the way we do trials, we wait forever to get a readout in most cases. And in oncology, it's shorter than some other things, but we need markers, better PD markers, to see if drugs are actually working in people. And so that we can get a quick read early in a phase 1 trial on whether something is working. And there are precious few of those types of markers. And I think if you understand mechanisms better, then you have a better chance to find a PD marker. But I think that area really merits investment.
If I might bring another point about cooperation between the academia industry, but it's just the general feel, is about, as new assays develop, we talk about immuno-oncology, there will be, more functional assays rather than relying on cell surface markers, for example. There is still a need for standardization if we're going to translate this, into drug discovery programs. But the other one is robustness and reproducibility. And I think we need to find a new way of doing science, both at that interface. And
along those lines, I think if there's a way for industry and academics, for that matter, to share negative data, it would really help, because that people try the same thing that others have tried, and they know that it doesn't work. So I think that would help the efficiency of the system as a whole. I don't have a solution for that, but I think that sort of thing would help.
Right. I'd like to expand on that point a little bit, and particularly to think about and get some guidance from the panel. What are the areas where people feel where there's an opportunity to accelerate early stage drug discovery in oncology and perhaps improve success rates? What are the technology gaps? What are the holes in your toolbox that you think would make a big difference to you? Is it in terms of translational models, better understanding, earlier understanding of the outcome of combination therapy studies, faster development of new modalities? What do you feel is holding you back from moving faster?
Maybe I can answer that question by also responding in the previous thread, because I think they're related, which is that I think there's a danger, given the cyclical nature of markets and the sort of burnout from crossover and public investors with the number of companies and the valuation of those companies. There's a baby in the bathwater problem that I think we need to continue making the investment in these explosion of platform technologies. Not all of them are going to be successful, but the worry is that with the market pullback, there's less investment in these incredibly exciting novel modalities, whether we're talking a resurgence of interest in covalent chemistry, chemical proteomics, molecular glues, degraders, et cetera. And the list goes on of very novel approaches. And there's a whole other list on the biologic side. And that we need to continue to let those platforms play out. They don't need to be public companies, but I think if we starve those platforms, we're not going to see the utility of those technologies in doing exactly what you were saying, which is accelerating the development of novel medicines, accelerating SAR cycle time, for example, or accessing larger target universe, which these novel technologies often do. So I think, I think we need to make sure that those platforms are given the time they need. Often that's at least 3 to 5 years to actually deliver these transformational medicines. So that would be my answer, which is keep smartly but efficiently funding those kind of novel technologies.
So actually, what Adam says really resonates. I mean, you mentioned Adam earlier on, AI and DeepMind and AlphaFold. Actually, the progenitor of all that was the CASP stuff that came out of academia. That started in 1994. So the vision that primary structure could lead to, 3D structures and there's still limitations. Started more than 20 years ago, yeah. And in fact, we talk about PROTACs. The first paper Ray Deshays and Craig Cruz wrote was 22 years ago, yeah. And glues, we came across that serendipity, including Celgene and Boston group, but that came out of the blue. It was unexpected, and now it's become part of a paradigm shift.
I think in terms of specific things that, advances and tools advances that can be helpful, I think we've talked about some of, just having really strong biological disease models that are more complex than, say single cells in vitro, right? Maybe they're organoids or I mean, this group can speak to some of the other things, but some certainly more complex models that take into account not just a single tumor cell, but the tumor cell in its complex, sort of much more physiologically relevant environment. I think we've talked about computational tools, and those are becoming increasingly biologically relevant, and they're building more, they're working more iteratively, right, with biological data so that they're tuned and improved that way. And I think that has to do also with sort of long-term advances in people being trained in computation and biology, right, which takes time to get there. And I think we're seeing that. And machine learning is sort of one of the computational tools, or sort of a computing tool. I think is being built. Those machine learning models are being built with increasingly larger data sets and higher quality data sets, and that's going to drive better ML models. And I think another area, and I'll throw this out there, and I'd love to hear sort of what my co-panelists think about these, but another area is really new views on cellular activity. And so, enhanced imaging, enhanced sort of microscopy, or other ways to derive insights on dynamic systems within the cell in disease and health, and how that can help us kind of crack open.
Katie, I'd like to really support one of many of your points, but one in particular, that's the rather mundane idea about models. We really, in oncology, we really need better models. I mean, everybody uses mouse models and they're just not very good.
Yeah, I couldn't echo that more. I mean, immuno-oncology, I don't think we've got any models that are predictive of any sort. We all use the same B16, F10, but we know its limitations, and yet we still carry on using that as a paradigm.
Although I have to say that so completely couldn't agree with everything Katie, Raj, and Rusty you were saying. I do wonder if, and this is really more of a question rather than a statement, but I do wonder if perhaps we're not getting the more creativity and the models and the tools, as much interest in development of those tools. And a lot of the times investors, I myself included, we don't like to invest in med device, we don't like to invest in services, and we don't like to invest in diagnostics or companions. I do wonder, perhaps it's something that could start on the earlier side, for example, through grants, perhaps, as you're saying, more grants, it could be done.
I agree, but this also goes down to grant panels. Yeah, some of these experiments that we're talking about, like getting the culture conditions, co-cultures, It's talking about serum conditions and right median, timing of passage. Why do some patients' pediatric, organoids grow, others don't? Pre-treatment variables, all of that. That's not glamorous. That's not going to get you the paper. Right. It's not going to get you the sort of podium talk at ACR, but it is necessary. So we've got to make sure that our governments fund that. That is where that interface of translation. I can't see investors doing that at this stage, given our timelines.
Thanks. I'd like to change tone just a little bit for a moment and really come down to understanding, what do you think the headline newsmaking technologies or progressions in the field are going to be over the next 1 or 2 years? Perhaps Adam, you'd like to start.
Sure. So you're asking to prognosticate here about the future, which is always dangerous. I would say there are two areas. One is sort of more process oriented that I think would significantly accelerate the business that we're in. And then on the scientific side, I would say from a process perspective, what we really need and I think are emerging and I think will significantly accelerate cancer drug discovery and development will be further democratization of clinical trials. I think this is a real bottleneck. There's certainly a lot of companies and very few patients actually benefit from clinical trials. And so I think there are some digital tools that are emerging and other approaches that will make clinical trials more accessible to patients and then therefore make clinical development and accelerating the transition to the market much more effective. I think on the scientific side, I think we'll begin to see the emergence of cracking the problem of cell therapy and solid tumors. I think that will come. I don't know what the timeline is. I'm not going to say go that far, but I think that's going to be a significant area of investment and also changing the targets of being undruggable. I think eventually every target will be able to be targeted by a small molecule or biologic. And that's what I look forward to. I don't know how long it's going to take, but hopefully in the near term for patients.
Thanks, Adam. Rusty, would you like to make a point here?
Well, I think for true innovation, timing is important. I keep remembering that the fact that I worked on, my colleagues worked on gene therapy at Chiron in the 90s. And finally, there's some inroads being made in gene therapy. So our timing was off. And so when you think about innovation, you have to be, just beyond the edge, but not too far out. And so I think Adam did a good job of summarizing some of the key areas where that timing might actually happen. I like what he said because of my own interests about a second 2.0 version of immuno-oncology. I think that and cell therapy, not just cell therapy, but other approaches to immuno-oncology, I think that there's a lot of promise in that. And I think that I also agree that having better designed clinical trials, if we could do that with biomarkers, as I said, the PD markers and patient selection, that'll help a lot. I don't know which therapeutic entity is going to, be featured at ASCO or ACR or SITC, but I think I think there are a number of candidates, and there's really I'm really optimistic that things are going to show progress in the next few years.
Raj, I know you work very closely with the Francis Crick. You must see a lot of exciting fundamental science. Do you see really groundbreaking new discoveries coming out of that in the near term?
Yeah, I mean, you've talked about 2 to 3 years. So, I mean, I think PARP inhibition and synthetic lethality was a great Steve Jackson, Alan Ashworth, labs. I think we're going to now see DNA damage response exploding. Then 2-3 major companies around this, but it's still very narrow, and so. And certainly the crick is playing a big role. Simon Bolton, set up RTOS, etc. in the UK around that. But it is now extended to nucleases, other sort of targets, which I think is going to make a big difference. But I really think the immediate time, I look at protein degradation, molecular glues, not just this drug discovery, potential to create drugs, but they are in many ways the sort of chemical equivalent of RNAi and CRISPR. If you've got a library that can degrade proteins, you can use them for the sort of phenotypic screens that Katie talked about, imaging, etc. And in partly Monte Rosa is built around that, but there are other companies emerging in that field to really exploit new libraries. And I do think we should not forget the phenotype. And historically, phenotypic screens have yielded great drugs, and once I agree with targeted approaches, target-driven phenotypic screens, I really think with isogenic pairs of cell lines, again, is something that's happening in a number of institutions. But I think all of these technologies are there and will deliver in the short term. We do talk about AI and etc. and novel approaches, but I do think there's a lot of optimism, even in the short term, and we'll see major changes in that context. Yeah.
Okay. I guess a follow-on question to this, to the panel as well. If we were to regroup after a much longer period, 10 years for the sake of argument, what do you think we will be discussing then? And Adam, perhaps a brief comment from you first, but I'd love to hear from the others. You spoke a lot about precision oncology 2.0 in your language. Many more targets, more diverse targets. Those targets will need to be approached in many, many different ways, different modalities. Can you see us solving a lot of that to come down to a much more tailored strategy, target by target, cancer by cancer, in the very long term?
Yes, I certainly can. I think what's been remarkable over the past 3 to 5 years has been this explosion of new modalities for small molecule chemistry. It was, on the small molecule side, and I know there's been an analogous effort on the biologic side. And I think what we're learning is, when are those tools useful? I think we now are aware of them and how to use them. There will be more tools. But I think learning when to use them against which targets, which binding pockets need this mechanism versus that mechanism, and matching the mechanism, the modality, to the pharmacology that you want for that target. I think that's where we need to do a lot more work, and we're beginning to learn to learn the rules. I think we're not quite there at the level of rules, but I think we can get there.
Adam, I couldn't have said it more. I actually was going to say something very similar, but I do want to also add to it. I do think that perhaps, and it's really more of a guess, but in 5 to 6, 7 years from now, there's going to be much more consolidation, because at the end of the day, there's a lot to manufacturing in the cell therapy, gene therapy space, whereas the actual explosion, I think, is going to happen more in the small molecule space in oncology. So I would watch for that in the next 5 to 10 years.
I think that moving from and hopefully seeing successes in all the more solid tumors, or in addition to the liquid tumors that we've been seeing successes in. We talked about cell therapies that came up along from Luba and Adam, and I think, in those, it's innovation, but it's also the costs, right? And so I actually would like to be I'd like to be able to say that, the costs are going to be brought down, through optimization of engineering and cell manufacturing processes. And I think we're seeing that. We're seeing, things as maybe they seem mundane, but really there is a lot of optimization and engineering that has to happen still. And then with that, hopefully we can get to a point where we're treating, the patients who can benefit from these kinds of, all the patients who can benefit from these kinds of therapies. So it's both, the cell therapy component, but also then really hoping to see a lot more success in solid tumors.
Rusty, do you think we'll see engineered B cell therapies?
It's one modality that has a lot of promise. I think training, the fact that when you use checkpoint inhibitors, you see that patients already have an existing immune response to tumors. Suggests that if you can enhance that response, you can get a therapeutic benefit and long-term immunity. And so I think that's a direction worth going. Dendritic cell therapy is too hard. That's the other cell type you could use to do this, but you can't get circulating cells that are the right kind of dendritic cells. So I think B cell therapy is promising. I just want to make one comment briefly about what KT said. The manufacturing is a big deal. It's true for all modalities, certainly for biologics drugs. It's a big impediment. It slows things down, waiting for slots, et cetera. But cell therapy, it's a gigantic issue. And people are working on this, so I think the cost will come down. And I think that cell therapy does have a lot of legs, and it will be useful in solid tumors. B cell therapy is focused on solid tumors, but I think other cell types will be used as well.
Thanks, Rusty.
The last 10 years, I'm looking forward to a post-genomic era, essentially. I think genetics has helped us understand tumor heterogeneity, evolution, the Sanger, have come up with mutational signatures, etc. But what that tells us is the problem. It doesn't give us solutions. And I know there's a drive now to understand drivers of heterogeneity, epipec genes, etc. I just do not believe that we can rely on 2 or 3 enzymes that have driven evolution over millions of years. And so maybe what we need to understand are the consequences. Why does a tumor cell survive just by having 50 mutations? And there is adaptation, and that will be at the RNA, the protein level, particularly, cellular adaptation. I think we went through metabolism, but it's going to come back. We just need to understand it better in context. And then immune escape is very much part of both how tumors have evolved and got away with it, and understanding those mechanisms. In 10 years, I think we'll do it, or we'll be partly on the way. So me, just to sum it up, won't be popular, a post-genomic approach to cancer therapy. I think the patient journey has to be different, and it'll be packages of therapies rather than specific, just one-off treatment based on molecular characteristics. And by that, I mean, we will be diagnosing patients earlier. We will be maybe even tracking patients and trying to do early diagnosis based on risk factors, which we understand better.
I love that. Just to follow up, I think at that point we really would understand and treat the whole person rather than the particular diseases, and understand also what are the comorbidities much earlier, and as I said, treat the whole person, but really with Raja's approach. And so I think there will be a lot more collaboration because we'll be thinking about outcomes ultimately for the person and the improvement of quality of life over long term.
I think we'll also learn along these lines to sequence therapies optimally. Do you do chemo first and then cell therapy or antibody therapy? Which goes first? Which go together? And what is the optimal sequencing? I think that'll be an important paradigm shift.
Thanks, Raj, and thanks everyone on behalf of patients waiting for new treatments for the huge effort I know you all put into making advances in this field every day. So thank you, everyone.
Thank you.
Thank you.
Thank you for joining us on day 2 of the WuXi Healthcare Forum 2022 for a compelling series of discussions on cancer, cardiovascular disease, and the neurosciences. Please join us tomorrow on day 3 of the forum, beginning at 8 a.m. Pacific Daylight Time. We look forward to seeing you there.